ingestion exposure of all these toxicals in there. So that was a kind of an add-on to the wildfires, which kind of neglected in the literature. So we never study. We study what happens during the fires. What goes up in the air, but whatever is going to be left on the ground, that is kind of understudied. So, and of course, we're looking toxicologically to understand what is the bioactivity. So it's kind of a study design. Yeah, I don't have time to go through this, but a few slides. This is a paper we're currently writing. The difference between a wood fire and a biomass fire, like the Canadian fire. Dramatic changes. This is, as you can see, this is the metal content. 10 to 100 times higher metallic nanoparticles than the Canadian wildfire I mentioned earlier. So this is, again, this is pHs. You can see for the LA, then the presence of both low and high molecular weight pHs. Compare the Canadian fire, biomass fire with LA, you can see in terms of the pHs, 80% higher than Canadian from the total pHs. But, and also 450% higher than other biomass fires in Southern California, and 7 times higher levels pHs that we usually study. So this is primarily because of the magnet structures that they don't burn, as I mentioned earlier. So this is ash. Again, we're analyzing the data as we speak, but so this is the aerosolization. We wanted to know how much is chemical. We collected ash in many locations, and you can see that, and that's the case across the board. About 75% of this ash is inhalable. It's less than PM10. And we're doing tox studies as we speak. Now in the lab also, because we want to study what components and what conditions of wildfires affect bioactivity, we developed platforms, maybe not as sophisticated as the ones that Pradeep has in his lab, but this is a platform that we developed with some money from NSF and VAS to study in a controlled way. We can burn different types of fuel, we can control temperature and other key parameters. We can control complete combustion, and we can generate particles that we can collect in size fractions. We can expose animals, and actually we just got an IRB approval and we start exposing humans as well, looking for cognitive function and some biomarkers for neurodegenerative diseases. So this is how we do things in the lab. So we don't have to sit there and wait for a fire to happen, but we can study in a controlled manner the bioactivity and the chemical characteristics. You can see we published probably 10 papers on this in the last couple of years. So again, I'm not going to go through the ongoing top studies related to the wildfires, but we're working on that front as well. So, and this is what I mentioned earlier, the wildfires, in terms of the pHs, what is really very striking is the presence of what we call oxygenated pHs, which are highly carcinogenic. As you can see here, we had benzo biciclone. So apology for going really over my time. I have to acknowledge it takes a village. My students, and I'm blessed to have a huge group of collaborators across the board working on this. So, and we are hosting in New Jersey a symposium in May 6 on wildfires, so you're all invited. So this is Harvard and Rutgers are co-hosting this symposium. We're going to have the NIHS director to be there to debate and study the known unknowns and the unknown unknowns related to the PM. So, and we're hiring, so we have some like some new grants. So if anybody in the audience or beyond has an interest in working with us, send me. Thank you, and again, my apologies for going over.
Thank you very much, and we'll have a Q&A later on. And next, I'm very honored to introduce my co-moderator for the next talk. So when we think about air quality health effects, you know, very often it's like a COPD and maybe heart attack, but how about your sleep? So the next presentation is about indoor air pollutants, sleep and air quality, right? Thank you very much.
Thanks, EY, and I think the former speaker set this talk up well. So it's impossible to get through indoor air pollution and respiratory health in 15 minutes. Obviously, the field has had an asymptotic increase in the amount of evidence. So the objectives of the talk, we're going to discuss the epidemiology of household air pollution and respiratory disease, and then talk a little bit about the interventional studies that have been going on over the past 30 or 40 years, and a little bit about the debate related to those studies. Here we go. So, as John mentioned at the start of the talk, exposure is very varied across environments. And so when we break it up between low and middle income countries versus high income countries, exposures related to trash burning, crop burning are obviously different from those of pollution, smoking in the home, as well as allergen exposures in inner city areas. And so the first part of this talk is really just stating that exposures are unique and different. You really have to actually identify individual exposure. You can't talk about this as a homogeneous term. Ambient air pollution is a leading cause of indoor air pollution. Obviously, everything that's happening outside comes inside, particularly in areas where there's increased ventilation to the outdoor areas. Tobacco smoke continues to be the leading cause of indoor air pollution in 50 to 90% of emissions in high-income countries. However, the area that I focused on, and most of us in the field are now focused on, is biomass exposure. So 3 billion people utilize biomass daily for cooking and heating. This is an example of 4 stoves that from a 13-site, multi-country site we did. But the type of emission you can tell, and the type of fuel is different and distinct. And so in Puno, Peru, they utilize dung for cooking and heating. In Dhaka, Bangladesh, we have wood. In Nagasaki, Uganda, we traditionally use charcoal. And so the type of exposure or the type of fuel that's being utilized is different. And as a result, the type of exposure that individuals that are exposed are different. This is a picture actually from Kirk Smith we were talking about Kirk last night. And so this is the first person to have their exposure monitored in the longest running activity, which is traditional fuel cooking. And so Kirk went out to Gujarat, India in the 1980s. And he put an exposure monitor in these older pump filters that I think John was mentioning. And found that this individual was exposed to something to the equivalent of 10 to 12 cigarettes per day, just cooking, doing that daily activity, morning, afternoon, night. But we now know that exposure, there's some debate as to what is that individual's actually inhaling and the particle size and toxins that individual is inhaling. But when we think about a important public health intervention, perhaps the single most important public health intervention, it is around this idea of exposure mitigation for cardiopulmonary disease, morbidity, mortality. And these exposures result in a range, or rather these fuels in indoor combustion results in a range of exposures. And just to show of hands, actually let's do table number 1. How many million Americans do you think utilize wood or charcoal for cooking or for heating in the United States? Just a guess. 30 million? A little high. Back table. Yeah. So it's about 6 million Americans, right? So this is an issue here in the United States as much as it is an issue globally. And there's a range of deleterious health outcomes. Lung function begins in utero, and so during the 4th or 5th week of pregnancy, our lungs begin to develop. Acidic or really early branching or airways begin to develop by the 13th and 14th week. And any exposure during that time period, either as a result of smoking during pregnancy, exposure hypothetically to biomass, result in lower lung function trajectories. And our lung functions continue to increase over time up to the age of 21. And so some people are getting close to this period of time where our lung function starts to decline. But if they begin low, we can see that these trajectories potentially result in lower attenuated, lower lung function gains in adulthood. And these are the individuals we're concerned about. So an individual that has lower lung function at birth has a 40 to 50% chance of developing COPD in adulthood. And this has been demonstrated in multiple studies. And so when we think about population and health, and when we think about exposures across the lifespan, starting intrautero at the age of conception is perhaps one of the most important time points. In terms of respiratory tract infections, there's a 78% increase in the risk. This is a meta-analysis that's done by a number of studies and analyses. 78% increase in the rate of pediatric respiratory tract infections. And when you think about intervention studies, the reason why they're utilizing this as a primary outcome is because of the results, but also because it is easy to ascertain when we think about a short period of intervention as opposed to longitudinal lung function interventions. There is a dose-dependent response for ALRI. So for every 10 microgram per meter cubed increase in PM2.5, there's a 15% rate increase in odds of respiratory tract infections in infants. The data on asthma is a little bit more mixed. And so there's some studies that say asthma increases. We have obviously more studies in the United States. Some of the work actually out of LA and on the West Coast have demonstrated the increase in ambient air pollution results in increased asthma emissions. But we do know that there's a relationship between secondhand smoke that's been very well established, as well as a hypothetical association between hospital air pollution. Nitrogen dioxide has been tied to a lower peak emissions. And lastly, COPD, which is my interest, and we get a little bit more into the details here. So this is a graph really about the distribution of exposures related to COPD across the world. So we have high income settings that are going to be lighter colors, lower income settings that are going to be darker colors. And we see how exposure and risk related to COPD change. Globally, tobacco smoke remains the plurality of risk related to COPD. But we can see how that changes 74% down to 29% in lower income countries. All at the same time, indoor air pollution, household air pollution, occupational exposures begin to increase. And so the risk profile really changes by country and by income settings across settings. And there's some interesting relationship between lower lung function or airway branching and furnace susceptibility. And so this is a newer line of investigation in pulmonary medicine, something called airway to tree branching. And so you can see an individual has good branching all the way to the periphery of the lungs. This is an individual that has dysynapsis or lower branching. You see a loss in the right lower lobe. John Verbo's group out of Canada took a big population-based study and looked at air pollution. He found that individuals with dysynapsis or lower branching in these areas were having increased risk of lung function decline by about 100 cc's per year, and then an 87% increase in the rate of COPD exacerbations. And so this idea that exposure happens at one point in time and sort of makes you susceptible to growth, it's probably not true, right? Exposure is cumulative. And it begins in your urine and it goes all the way to adulthood, even after a point when you have COPD. Any pathologists in the room? Good. Sort of get everyone saying anything. So this is necropsy results from Mexico. And Rivera and colleagues have been thinking about this for a while. And it's interesting because I do COPD all over in high income settings and low income settings. When you go to low income country settings and ask the physicians, what is COPD? It's very much described as a different disease than what's being written about in the literature. And the reason why is because most of the clinical trials that we do today, and certainly most of the epidemiologic data we have from COPD involves individuals that have smoked 10-pack year smoking history in high-income countries that do not have any close to exposure in low-income countries. And so low-income country investigators for years have been pushing on this concept that perhaps the disease that we call COPD is distinct and different based on exposure. And so Rivera and colleagues basically did an necropsy from women cooking with wood stoves. And this is cigarette smoke, biomass smoke. This is all predominantly wood. And we see a different pathology. Right? Again, this is sort of a series of cases, and so you can take it with a grain of salt. But there's increased goblet cell hyperplasia, cigarette smoke as opposed to biomass smoke. There's increased pigment deposition, so this is anthracosis, both in the airways as well as the of the pulmonary vasculature, and also we see increased pulmonary vascular thickening. And then, the COPD studies in low-income settings really are about 10 to 20 years old. As we're following these cohorts out, we actually realizing that they're presenting with different phenotypes or different presentations of disease. These individuals that are exposed to biomass smoke tend to be a little bit less hypoxemic, but have higher rates of mortality, have worsening functional status. And so the field is now moving towards this understanding both exposure, but also the disease and pathology associated with that exposure. As CY mentioned, an increased area really in the field right now is related to sleep and sleepiness. And so PM 2.5, nitrogen dioxide, and associated aberrations in the target circadian rhythm and circadian cycle, as well as obstructive sleep apnea. It's thought to be a result of a range of pathophysiological phenomena, including this lack of control of essential processes of sleep, as well as increased inflammation of airways, resulting in collapse of the airways in sleep. But obviously, two things that we do, we're all exposed to something in our day-to-day life, we all sleep. And so when you think about exposure and disease relationship, it is certainly an area of interest in investigation and potential intervention. Now, the pathophysiological mechanisms related to exposure, were elucidated in this lecture prior to, but they range everything from macrophage activation to presenting antigen, including our response or our inflammatory response to exposure, including oxidative stress, apoptosis, pulmonary surfactant deactivation, reduced mucociliary characteristics changes. And so there's a profound inflammatory cascade that occurs when your body is exposed to either particle from firefighting or wildfire or cooking stove. Now I'm going to talk briefly about exposure reduction. And so this concept of how we reduce exposure is certainly not novel. It goes back to the 1980s. And it is certainly one of the first things Kirk Smith thought when he saw this cooking visual. And so weak exposures are numerous. And so the ones I'm going to focus on are PM 2.5, the rest of this talk, but nitrogen dioxide, allergens among those individuals susceptible to asthmatics, molds, and of course environment, tobacco smoke, which in high countries is perhaps our most important intervention. This is one of the first and most important studies related to respiratory diseases. So this was allergen mitigation on individuals with asthma. This was done in 2004. And this was a very complex study. So it was a 12-month intervention that included allergen mitigation, including pesticides for individuals with cockroach exposures in the household, HEPA filters, we did mattress covers, education for children and families in inner severe areas. And for those that sort of understand Kaplan-Meier plots, this is the maximum number of daily symptoms, and you see controls over the course of the intervention group. Over time, there's really a immediate drop. For all primary and secondary endpoints, as John mentioned, p-value less than 0.05, but this was really a consistent phenomenon. For those of us that treat individuals with asthma, this is very, very, very difficult. Right, so outside of a randomized controlled trial, doing all of these interventions in our populations, it is tough, particularly among those individuals that are susceptible to individuals that are in settings where these allergy exposures are common. Now, particular matter is really taking center stage because when we talk about those 3 billion people that utilize cooking daily, going back to the 1980s, the thought obviously was, why don't we improve the stoves? Why don't we reduce the exposure in order to improve the deleterious health outcomes that is presented? So this is something called a rocket stove. So in the most fundamental setting, a cook stove basically aims to change your exposure as a result of an open flame, 3-stone open flame. And so this basically improves fuel efficiency, it decreases the amount of what necessary intake, and hypothetically, it reduces exposure to the individual that's cooking. This is an LPG stove, and so for a number of reasons, which I'm going to get into in a second, we've sort of moved over to LPG stoves. But there is robust evidence really for the former, increasing evidence for the latter in terms of its effects on health. So this is a busy slide, again, going back to Kirk Smith, but the takeaway is no difference, no difference, no difference, no difference, no difference. Does that mean that these cook stoves don't necessarily work? The important component of this is none of these really resulted in improving the exposure reduction. Right? And so when we actually did personal monitoring on individuals, and Hawkins trial was perhaps one of the best with personal exposure monitoring, we didn't really reduce exposures to a meaningful level. And so while some of these studies resulted in improved efficiency in terms of food use, and some of these resulted in less wheeze, symptom quality, none of them really met their primary endpoint. For a lot of these, that primary endpoint, childhood illness defined. And so that led to Kevin Warner, John Moss, for 2 very, very advocates in the field. What's the next step? Do we continue with these cook-stove interventions, these lowest cost interventions in low-income settings, or do we do something better? And what was bigger was basically a multi-center LPG trial. So this was done with 3,600 individuals, dyads and mothers that were pregnant, as well as other individuals, older individuals in the household. They followed these mothers 2 terms of pregnancy and then followed infants. And when we think about it, again, an outcome that's easy to measure, pediatric outcomes are incredibly important, incredibly this study, unfortunately, was done during COVID, so that also complicated some of the follow-up and also some of the enrollment, so take results. But what it was very successful with was exposure reduction. So individuals in the intervention arm achieved a 66% reduction. About a third actually met WHO guidelines, which is actually phenomenal when we think about some of the environments in India and Guatemala where participants were enrolled. However, these studies came out about 2 years ago and were serial articles in the New England Journal, but none of them met or the same. So this is their columns were basically looking again at severe pneumonia outcome. We see it's possibly 1, and all the secondary outcomes were negative for stunting, for fetal growth. Those outcomes were also, which sort of leaves us now at an interesting place. I'm interested actually to hear John's point or John's comments when we get on the panel. Is this really the end of intervening for clean cook stores? Is there meaningful outcomes or are there meaningful data that we can gather from this despite achieving over exposure reductions? I will say that the concept of exposure reductions is certainly not. And so my hypothesis is that in the right population, the susceptible population, we will see some. So this is data from Nottingham Ancestry group, and this was a study with 100 people, right? So another study had 3,600. This is 100 individuals with COPD, and they utilized indoor air filters, rapid filters to result in air purification. We see meaningful exposure reduction between the intervention and control, both 3 months as well as 6 months, and they met their target for primary outcome, which was essentially monitoring exacerbations as well as decreased use of rescue medications. And so I'm going to stop there, right? There's a whole litany of research related to exposure and exposure reduction. But what I will say is there's robust evidence to support the association between household air pollution and range of cardiovascular outcomes. The interventions, though, however, are mixed. And I think a combination of this is really understanding the populations we're targeting, but also understanding the clinical outcomes we're seeking in that population. A lot of these studies, and John's certainly aware of this, are longitudinal studies. And so when we look at populations in China where they follow populations out for 7 months, we actually see decreased lung function attenuation. We're going to see more of that data, particularly as they do the long-term outcomes, long-term follow-up to happen. And then identification of exposures populations is paramount to understanding the efficacy of the intervention. So I'll stop there for the last talk.
Thank you very much. So we have heard talks from the medical, the public health, epidemiology, and now we're going to have one from environmental science and management. So if you wanted to mitigate fire risks, how do you actually do that? We cannot rely on medical doctors to do that, right? So we're going to have some people in the field to do the prescribed burning. And so this is for our next talk, Dr. Holly Noel from Toll Timbers, who is going to share her experiences. Please join me in welcoming her.
Toll Timbers, we're located in Tallahassee, Florida. We are a nonprofit research station, and we study prescribed fire. We started in the 1950s. We look at all things that are affected by fires that are set on purpose. So we started out doing fire ecology, which are plants that thrive on fire and have to have regular fire to bloom and grow. Then we also have Stoddard Bird Lab, birds that need fire. They need it for their environment to keep it clear. We have Game Bird Lab, which I'm going to talk a little bit more about in my talk. We have fire effects. What are the best ways to set prescribed fire for your management objectives? And in the last year and a half, I joined as the smoke science program because all of this fire involves smoke and we need to manage smoke as well. So I'm talking today about the fire in the southeast. So I'm going to do a case study on Albany, Georgia. But first, why do we need fire in the southeast landscape? Well, all of these cute animal pictures, which are always very helpful in a talk, all of these animals and the longleaf pine, they need fire. So for example, the indigo snake and the gopher tortoise, the frosted elfin butterfly, the red cockaded woodpecker, they're all threatened and endangered species. So they need the prescribed fire landscape because they like to have cover from their predators and to move around, but they can't move around if there's too much woody encroachment on the ground, if there's invasive plants, they'll die and they'll move out. We even have gopher tortoises on our property that something's changed in the landscape, even though it's been burned, and they've moved out of that area. They no longer have their gopher tortoise dens, which are super important because all these animals can actually take shelter in these gopher tortoise dens. So it is this really diverse landscape. And then you have the longleaf pine. The longleaf pine is actually a very important pine tree for this region. It used to occupy this whole entire area from like almost Virginia all the way out to Louisiana. And now it only occupies 3% of its former extent. And why is the longleaf pine important? Well, it turns out the reason why it's here is it's really good at standing up to hurricanes. It's really good at being not susceptible to drought and maintaining its root system. And with global warming and climate changing, it turns out that's really important. So a lot of it was cut down to make way for production pine for timber industries. And so now there's a push to reintroduce longleaf pine, but to do that, it needs regular fire about every 2 years. Also game habitat, as I mentioned. So the northern bobwhite quail is a super important bird. And I'll talk about that in a minute, and wild turkey. And then, of course, wildfire fuel reduction, hazard reduction. So you hear a lot of people saying, well, we're not like California because we burn all the time. We also have different landscapes, but that's a talk for a different day. But it does reduce fuel loading. So now annual average PM2.5, earlier talk set this up really well for me. But the annual average used to be 12 micrograms per meter cubed, and recently it was lowered to 9 micrograms per meter cubed. EPA has been talking about this for a while, and this map right here looks at a couple of different standards, but this shows all the counties using data back from 2019 to 2021. What would those averages be? And what counties would be in trouble if we lowered it to 9 micrograms? And you can see the state of Georgia had several counties that would be out of compliance with this lowering according to this earlier data. And working with the Georgia Environmental Protection Division using data from 2021 to 2023. So in case you didn't know, when we talk about annual average, we're actually talking about 3 years averaged together, but it's not talking about one single year. So that's why it has multiple years. These are all the areas that would be out of compliance, shown in red, borderline areas, or in yellow. And the ones that would be okay are in green. So each one of those blue dots is an EPA monitor that's being used. And so not only is it just the counties with the monitor, they have to include the whole airshed. So everything that would impact it. So they had a lot of areas that would be out of attainment. So now you can do something called exceptional events according to the Clean Air Act with the EPA. And exceptional events, they're unusual or naturally occurring activities. So this includes wildfires. So you can't control wildfires. We can't tell Canada we don't want their wildfire smoke that comes down here anyway. And the EPA recognizes, hey, that's not fair. You can't penalize somebody you can't penalize a city for not being able to stop wildfire smoke. So you can file an exceptional event. And that goes with not being reasonably controllable. And what you do is you submit a filing with the EPA. So you gather all this justification documentation. It has models. It has everything that you would need to show that, hey, this event did impact our monitor. Historically, it looks like this. We had this event, and now we're up to this. Then you have to open it up for a public comment period. So anybody can comment. You get organizations commenting. You get law groups commenting, environmental groups, and some interesting comments that occasionally slip through from the public. And they have to respond to all of those. So the EPA has to, or the local state agencies for air quality, they have to write responses, and they submit that all to the regional EPA who reviews it. They submit it to the national EPA who has to review and approve it. And then if they agree, that day doesn't count in your calculations anymore. They agree that it was an event that you couldn't control, that you shouldn't be penalized for it, so it'll get kicked out of this annual average. And they also say in the Clean Air Act that prescribed fires can be an exceptional event because they are used for land management purposes that I already talked about. However, this is an extensive and time-consuming process. So for example, in the state of Georgia, they determined based on these values that they would have to file over 151 exceptional event demonstrations. And what that means is a lot of paperwork. To date, although there's some under review right now, there's only been one successful exceptional event demonstration that was filed, and that was by the state of California, who burns way less prescribed fire. It was for a single prescribed fire, and it was over 150 pages of documentation. So of course, the Georgia Environmental Protection Division, they didn't get extra money from the government to do this paperwork. They had to use the staff they already had. So it was a particular burden on them. But even beyond that, you know, that's a lot. It's a lot of fire. So prescribed fire is used heavily. I mean, some of these are wildfire events, but if we're setting the prescribed fires, we could do better. We can control when the fire is set. We can see the meteorological conditions. So we can improve this. So not only to relieve the burden on them, but to relieve the burden on us because we live here too. So that's where we came up with the Albany Pilot Project. And so Albany is this county down here, or this set of counties down here. And they, at the time, were sitting right on that bubble of potentially being out of attainment. So why did we choose Albany? It's that bird right here, the northern bobwhite quail. And you may be wondering, what is such a big deal about this little bird? Actually, Albany is kind of a small community. It brings in 1,000 local jobs. So people like to hunt this bird, basically. So it can bring in $145 million a year to the Albany economy. So for example, if you manage your lands well for the northern bobwhite quail, you have hunting dogs that are highly trained, you have horses that are highly trained because they don't want to go on ATVs, they don't want to go on trucks, they don't want to disturb the quail, they don't want to disturb the landscape. Some of these areas that they will give hunting leases to, they can charge between $10,000 to $20,000 for a half a day of hunting. And people will pay it. So it brings in a lot in a short amount of time. And if this would go away, if the bobwhite quail would go away, there would be one paper processing mill that would be left in town, a Procter & Gamble, which is a different discussion for pollution. And also, when you manage for bobwhite quail, you're also managing for all of these. So the local land managers, they love to brag about how many gopher tortoises they have on their property. And that comes with the indigo snakes. So it's contributing in other ways. They have longleaf pine, they have these well-maintained landscapes. They have endangered species. A lot of them are recipient sites to receive them, which means, they've proven that they've got great land and they can prune them. So it's a very powerful bird. And so when you start talking about, hey, we have this issue with smoke, we can shut down burning, people panic. They don't want to lose their town. So it's a very visceral reaction from them. The people that manage this, they've been there for decades. Like, I talk to land managers there all the time. You've got a man who's in his 30s burning. Down the road, his dad is in his 60s and he's burning a different land. He's a different land manager. And they took it over when his dad retired. So you have a lot of people that are very invested in this particular landscape and their jobs. And they burn approximately 150,000 acres per year just from the months of February to April, because that's in between nesting season for the quail and hunting season. So the Albany Pilot Project was a collaboration with the Georgia Forestry Commission and the state of Georgia. If you want to you don't have to do it for agricultural burns or if you're Department of Defense. But if you want to burn in the forests or silvicultural burns, you have to get a permit from them. And they'll only grant it on certain days when conditions are right. And we work with Pineland Plantation, Albany Quail Project. Albany Quail Project is a branch of Tall Timbers, but they're land managers out there. There's a big 9 land managers in the Albany region. Pineland is one of them. Jones Center, which is a similar organization to Tall Timbers. It's like our sister organization. And literally, my 3rd day on the job, I was sitting at a table with all of these people trying to come up with what are we going to do to help manage our smoke, to get it below compliance, to make it healthier. And we came up with this second iteration. There was a Albany Pilot Project 1.0, which was not as successful. But we said, okay, let's just look at the 30 miles around Albany. So in this map right here, not only do you have private properties, you have the Jones Center, which is right here, which is another research station. You have lots of conservation easement properties. So they've signed their lands over to USDA, and they manage their lands for conservation purposes and perpetuity. So they have to have fire. And we said, okay, how can we make this better? So here is the center of Albany. It's where the sensor is. And if you know anything about fire weather, smoke is moved by transport winds. So if you've seen prescribed fire, you can see it. This doesn't happen all the time with wildfire, it's too energetic, but sometimes you'll see it stop and then just like go out. So that top layer where you see that smoke stop, that's called the mixing height, or the boundary layer, some people call it. Transport winds are the winds from the surface all the way up there. That's what moves smoke out. So let's say we have transport winds coming from the southwest. That means anything that the Jones Center and these properties right here burn are going to move right over Albany, the biggest population center in this region. So we said on days when that's going to happen, there's going to be some additional criteria. We got to make it a really great smoke day. We have to have that smoke move up and out. We don't want it to hit. So if you're within this cone area, when you try to call in and request a permit, you're going to have to make sure that there's not a lot of PM hanging around from the day before. They were looking at a 24-hour average. If your airship's already contaminated, no burning that day. Or if your smoke dispersion, which is a measure of how well your smoke's going to mix and be diffused and move out, is low, you can't burn that day either. And if transport winds are low, you can't burn again. And then it has tiered criteria depending on how high the transport winds or the smoke dispersion is. But everything, they wanted you to be done by 2 p.m. so that it moves out before nighttime settling. That's when we were getting a lot of exceedances and PM values are really high, is when the boundary layers sank at night. But if you're in this 30-mile region, it wasn't as strict, but still just some good rules of thumb. And honestly, these are things they should have been listening to anyway. These are just characteristics of a good burn day where you want good smoke dispersion. So did it work? Well, here is a figure, and all the gray lines you see right here, those are quarterly daily running averages. The EPA does this quarterly average, and they average the quarters together to get your yearly average. So typically, for each quarter, here's the quarterly averages right here down here from history. And then you'll get this annual average of 9.2 micrograms per meter cubed, which is slightly higher than the 9.0 we need. So it's right on the borderline. And doing the project for the whole entire year, the 2.0, it's currently estimated to be at 8.4 micrograms per meter cubed. So quarter 4 is still pending official results, but it looks like it was a success. Every quarter was below historic average, but I do caution everybody who starts partying in the Albany area that yeah, we did it. This is n equals 1, so it's not a statistically robust measure of success. And I think very important things to keep in mind is there are droughts in quarters 3 and 4. So there was not a lot of burning going on, and what's starting to happen right now is the whole time I've been here, I've been getting text messages from land managers saying they're mad, they can't get their permits because the smoke dispersion is really bad. And I say, well, that really sucks, but you can burn another day. It's only January, so people are starting to get anxious because the drought is ongoing. So we'll see what happens this year with the project. Also want to look at meteorology, because meteorology is just really special this type of year. How did it compare to history? So there's some more things to look at. But what we do see is that it did slightly alter bird behavior. Now I will say that, again, this is n equals 1, so none of this is statistically significant. But if we look in that 30-mile buffer in 2025, about nearly the identical number of permits happened, and they burned 5,000 more acres than last year and 3,000 more acres than 2023, where there wasn't the pilot project in place. So they were able to burn more acreage, and they had less annual PM 2.5. And they got the message there was less burning in the cone. So this is a hint of a change in bird behavior. And I will say that I have made a concerted effort. Part of my job is to go out and talk to land managers and to get them to buy in. There is a deep distrust of scientists. They are tired of people telling them what to do and how to manage their land because they say they get involved after the fact. So I've made a concerted effort to talk to them and make them part of the process. So trying to get them excited to learn about, hey, we need to start looking at what's actually in the smoke that, we're breathing in and you're producing, but I want you to be partners with me. I want you to be part of this research. And they're starting to warm up to it and come around to it instead of being insulted by it and trying to ignore it. So we have tweaked the pilot project a little bit for this year. One of the main complaints was that they were missing ideal burn days. The pattern that kind of happens in this area is that you have cold fronts that move through overnight a lot and it cleans out the air shed. And so when you're using that 24-hour average, it overlooked these farming passages. So your 24 hours average could be really bad. You get a cold front coming through at 3 a.m. and then you're sitting just watching this really ideal burn day pass you by. So we started looking, and it was also difficult to explain and interpret for the people that issue the permits. So we set this 6 a.m. value at 55 micrograms per meter cubed. It looked into some data that kind of indicates, if it's going to be a bad burn day or not. And that helps analyze with the AQI. So what's the future? We want to continue and monitor to adjust the pilot programs, educate private burners. This has been a big drive. So we have this class. It's a free class. I know it says it's not available right now, but it's called Be Smoke Savvy. Everyone can take it. They learn about weather meteorology and everything they can do to help manage their smoke. And we have targeted media campaigns, liveprescribedfire.org, explaining why everything's happening, what people that are exposed to smoke can do, who they can contact to find out when burns are going to happen. And just introduce better tools for smoke management. One of the other projects I'm involved in is called this Prescribed Burn Planner, which is free. They can put in their ideal meteorology days, and they'll get an email that's like, hey, you got some great burning days coming up. Oh, hey, you don't have great burning days coming up. They can put in all their properties. So they're maximizing great burn days and not missing good burn days. So I can take questions in the panel.
Thank you very much. Now I'm going to ask the panelists to come forward over here. We're going to start the panel discussion. Okay, I know there are a lot of questions from the presentations, but being a moderator has a privilege. I'm going to start with the first one. Okay, so for the panelists, I would like to ask your opinion. How can interdisciplinary collaboration between atmospheric scientists, public health experts, medical experts, policymakers act as a solution for mitigating health impacts of fire emissions? I want to start.
So, has anyone been involved in policy change? Okay. So it's really interesting. You probably all know this, that policy change is very rarely based on empirical evidence. Very rarely. And in the state of Florida, when we work collaboratively with the firefighter unions and others to try to get presumptive legislation passed in the state, which means that cancer is recognized as a line of duty illness, no one was actually really interested in the science, which is unfortunate. Everybody was really interested in the impact of the policy change on the pocketbooks, if you will, of the municipalities that are ultimately the employers of firefighters. That said, we cannot solve an issue like the impact of wildland fire on health without having legislators at the table and having the benefit of interdisciplinary collaboration. These are complex issues and they require people to feel comfortable working across their epistemologic boundaries to figure out how to ask the right questions and how to layer findings in a way that advances knowledge on multiple fronts simultaneously. And so certainly Sylvester's Firefighter Cancer Initiative involves all of the various different scientists that you mentioned. I'm a population-based scientist. I work closely with Cassie Gaston, who's an atmospheric scientist. We think, we try to find common language by which to communicate. It's not always simple for me to understand what she does, but it's important to have that tenacity, because if we don't work in an interdisciplinary manner, we're going to fall short of the expectation that we have to science, and also that legislators will ultimately demand in order to inform policy.
Thank you, Eric. Well, I mean, as part of the risk assessment process, we need to generate the data that the policymakers they need. So you got a feeling of the complexity, studying exposures. To assess risk, you need to have the exposure assessment, the characterization, the complexity of the chemistry, the aerosol science, etc. has to be addressed. We need this kind of data. And we need to go from the, sometimes, to assess risk also, you need hazard characterization data. You need the toxicologists to be involved to understand a little bit the bioactivity across biological systems. And of course, we need the epidemiologists as well, so in the epi studies to point to certain diseases. So that's true not only for wildfires, but any environmental pollutant. You need to bring all the disciplines on the table and go all the way from the start, molecular level and then exposure to population level to assess the risks. And when we assess the risks, then we can manage the risks.
So I hate the question. And I'll tell you why, because we often say these sorts of things and then talk about it. And I'm going to pull this out of the abstract. I showed you the diagram of how we revise the National Ambient Air Quality Standards. In fact, it does involve these interactions. And here, it's actually one of the cases where there's almost lines that can be drawn between science and change in policy. And those are not common. The question of how interdisciplinary groups actually work together and can combine and talk to each other is really complicated. And sometimes it works, and sometimes people sit in their own silos. And sometimes as a public health scientist, our silo is particularly looked down upon. I think probably many of you have been there. So, I think you need a framework in which things will work. And it's clear what the objective is. COVID is actually a good example because it brought together the aerosol community, the building folks, the infectious disease folks, the epidemiologists, and the policymakers in ways that had not happened around infection. I mean, there have been literally no advances in sort of airborne transmission since Wells Wiley about 100 years ago. So their necessity forced the interactions, and I will say that that can happen. But having sat around the table in so many committees where the engineers are talking to the engineers and the toxicologists are talking to toxicologists, you get the point. And trying to bring the science across towards some common objective is challenging. And I think having attacked the question, I'll say it's a good question, but that it opens up a lot of complexities about sitting in. I've chaired multiple interdisciplinary academies at the National Committees, National Academies, and elsewhere. And it really takes a while for people to learn to talk to each other and to appreciate what each brings to the table. But it can work sometimes.
We hired a media literacy group, and they went and actually got people at the grocery store and gave them $50 gift cards to answer a bunch of questions about smoke and prescribed fire. And surprisingly, in Albany, they didn't really know much about it. And they're like, oh, well, this is horrible. We don't want any fire. And when you tell them the benefits of fire and then what it does for the economy, then they're concerned about it. They're like, well, we want to know more about health. We want to protect our health, but we see the benefits, and then they call their representatives, and then all of a sudden now the representatives care and they want to sit down and they want to have a conversation with us. So it's really important, like you said, to address your message to a broad spectrum of people who have different interests.
So I actually thought Holly was going to say something different. So we talk about interdisciplinarity, but if you really want to have translational impact, you cannot forget the importance of community engagement. Like, we cannot do science that's separated from the people whose lives that science is intended to change. I'm sorry, we cannot. And over the course of 20 years, I've worked with many different stakeholders who are really well poised, despite limited formal education, to nominate research questions of public health significance, and then to think about how what their experience may translate to important work at the bench. Something like the Firefighter Cancer Initiative does not happen without firefighters who are working alongside us and helping nominate really important questions that are catalyzed from their occupational experience day in and day out. And then their ability to also speak about certain exposures that occur upon the job and then how it impacts them personally. There's sometimes latent hypotheses in what people share as part of their lived experience. And ultimately, we don't have translational impact if community members are not at the table in one way or the other.
So I think we were fortunate to see a wide spectrum in the short period of time of the data that has informed our understanding of the epidemiology of the environment. What are the critical gaps that remain in understanding fire combustion and respiratory and other long-term health outcomes?
Well, I think I mentioned it in my talk, the emerging sources of carbonaceous aerosols, they bring another layer of complexity. We have compounds that we didn't have historically out there, and the EPI studies were done based on historic EPI and tox data. So, but we're very lucky today. We have all these tools, medical tools, that we can really pinpoint and identify and characterize the complexity, which we didn't have before. So that's one thing.
So I'll make a comment based on PM experience. So in 1997, of course, the PM 2.5 standard was enacted, and it caused a great deal of controversy. And the Congress mandated that the National Academies would set a research agenda on particular matter that would address key uncertainties. And Bruce Alberts was the president of the National Academies, called me the day before Christmas. I remember still 1997, and asked me to chair this committee, and said, oh, by the way, the report's due in 3 months. And what we did, which I think is useful, was to set out a clear framework to identify the key uncertainties. And in many ways, the same framework applies actually to the wildfire smoke. We were in the unique position that actually both the House and Senate science committees were following our funding recommendations, and EPA was constrained to actually follow them. For the 6 years of the committee, we actually were able to direct funding, a lot of it which went to try and identify those characteristics of particles that were important. So I would say that getting organized around identifying the key uncertainties, and those might lead to something actual, was critical. And doing that same sort of exercise, I think, with wildfire smoke and those exposed would be a useful thing, and I'm not sure it's been done.
It's not as bad as wildfire smoke. Well, citations are needed for that. So we don't exactly know what's in the prescribed fire smoke. We don't exactly know what the health impacts are. we're relying on studies that are based on wildfires. So I think having something specific for prescribed fire so we can start doing better. Like, I made that pilot project the third day on my job. Like, I had no data to back it up. It's just what I know about meteorology. So I think there's a lot of work that needs to be done for prescribed fire in particular to fill in those data gaps.
So, quick comment, just to remember, and you're probably familiar with the Hill criteria for causation, or some of you may be. And Sir Austin Bradford Hill wrote this famous paper in 1965, and I think just an important point is he basically said all knowledge is incomplete, but we still have to take action. And I think sometimes in our quest to write the next grant and saying little is known about whatever we're going to write the grant about, we exaggerate. And I think that sometimes this is really critical to say, well, we may need to fill some gaps, but we can still take action, or we're going to follow the precautionary principle or something else in the policy context. And we can move forward, but we really want to nail down whatever it is, because that might be perhaps the role of respiratory protection during times of high smoke. Yeah, we don't need to wait for everything. So just a reminder.
Just a comment on the precautionary principle. I mean, in this country, unfortunately, for emergency prudence, we need to see people dropping dead and then we act. That's not the case in Europe, from Europe original. So, but I want to input with John. There is this meta-analysis done published in Lancet Prophet. You know that report on, it's not on wildfire, it's on chemical pollution in general. And it's a whole section on air pollution. We did an amazing job cleaning the air in the United States and the Western world, the global north. And we had the deaths from air pollution down to 3 million per year. But that report, which is focuses on the last decade, 27 to 2020, I mean, the number of deaths is going up. It's now 7 million people died from air pollution. So we went down and now going up. So I wonder your comment on this.
So you've got to use and read the global burden estimates with caution and true understanding of what they are. So the deaths attributable to air pollution reflect background rates of disease, which have gone up, along with the risk estimates for associated air pollution. Behind the global burden estimates are so-called counterfactuals. And so the 7 million is the world as it is with the distribution of particle exposure. If all particle exposures are reduced down, it's actually roughly 2.4 micrograms per cubic meter, which isn't going to happen. So there's a big difference between attributable and avoidable. The attributable has gone up in some places for a number of reasons reflecting underlying rates. So I think those numbers are really useful. You know, Tedros, the head of WHO, at the first WHO air pollution conference, said air pollution was the new tobacco, which I really didn't like. So this whole idea of attribution began with tobacco, where we could have a prevalence of 0. We're never going to have particle levels everywhere as low as the counterfactual, because it's impossible, both because of nature and because we burn stuff. So I would just say, be careful. And those estimates have sort of differing weight in different places, I think. And, you know, for example, when I work in sub-Saharan Africa, people are a little bit skeptical for the reasons I talked about.
So we'll pose questions to the group. Actually, yeah, I'll just do right up front.
All you happy, all your box is big. But vapors, we don't have a filter in our face when we are breathing. So you have particles plus vapors. Vapors are highly toxic. They can interfere metabolically, both synergistically and antagonistically with the effects of the PM. Why I have not seen anything on your API side of air policy interferes with vapor phase components. My Is it the complexity of the sampling and the sort of data, or is it the complexity of the compounds that are not included?
You know, we can't study everything all the time. And, you know, I appreciate your point, and somebody else could raise their hand and say, well, you're not studying this. And, you know, us poor epidemiologists, we deal with sort of the exposure measures that we have. And I appreciate this. A long time ago, we actually were working with one of the original PM Center applications was Paul Leroy, and he was quite concerned about this. I mean, you know, some of the issues with vapor phase components, for example, think about what people study in the lab. They go collect a particle sample, and everything that's volatile is not going to be in the particle sample. So it's a real problem to study real world mixtures. And, you know, people, you know, take their critters to the roadside, perhaps, which is one approach. You can expose people to real world mixtures. Folks in LA did that decades and decades ago, but for epidemiology, sort of having enough people with the kind of exquisite measurements of all the characteristic of particles or what's in the air, the complex mixtures, you're right, we don't do it. And I think it's sort of feasibility that is the constraint. And then I think ideally here we would have some sort of bridge from understanding of the chemistry to mechanisms of injury, that we could bring the epi and the chemistry and the tox together. And, you know, I mean, I think we're making progress, but probably not there yet. Maybe you want to comment?
Yeah, I mean, at least for mechanistic toxicology, that is embedded in the study. So I think we advanced our tool set in terms of studying mixtures, but we're never getting exposed to them polluting the time. So I can tell you from my own experiments, when we expose animals or even cells, now we're looking for synergistic effects from gaseous co-pollutants, which is the case in the wildfires as well. So, and also modeling has also evolved, so in terms of the source apportionment. So we can, so the tools are there, and there's also from NIH and encouragement to study mixtures of pollutants because they're always adding these synergistic effects. So there is a progress. Epidemiology is a little bit more complicated, as John mentioned, to have the exposure data for mixtures and develop the models. But there is progress, so it's not that we're studying.
Such an amazing and insightful presentation. My question is specifically to you, Dr. Philip. Pardon me, I don't know the last name, but then any of you two can also answer. So, as an up-and-coming PhD student or a PhD candidate, how do you deal with issues regarding generalizability, especially combustion studies in the lab? I ask this because I put in a grant and I was marked down specifically because of generalizability of my results. But then we all know the unpredictable nature of wildland fires. And so sometimes it's cumbersome or more or less impossible to gather your tools and then be there to collect data. Issues regarding prescribed bans, it's expensive, there's bureaucratic issues, and sometimes as a PhD student, and looking at the funds involved, you're unable to also involve and prescribe bans. And so when you want to do a study that has to do with the lab, you don't have animal exposure. Really looking at what you want to do, your only option is to do a combustion-based study, and then you are having issues with, or I mean, reviewers are coming at you because of generalizing your result. How do you navigate that so that you don't feel so sad and beat yourself up when the grants you put in there are being rejected on basis of that? Because I feel lab work is the first step of taking results and implementing or even building up prescribed bands to validate all these things, or even for those who are involved with modeling, sometimes you need data from the lab. And so if they are not giving you the chance to even start there, how do you manage such issues?
Yeah, that's a very good question. And managing complexity is not easy. I didn't have much time on my presentation. I had a special section on how we address these questions in using lab-based systems. So it was, I showed a few slides at the end, but in terms of wildfires, that's why we develop a lab-based, what we call wildfire simulator, which enables us in a controlled way to study different important factors of the combustion. And what does it mean, not only in terms of the chemistry of the particles, so you can burn different types of fuels. You can get prescribed fires fuel that we can burn, and we can also play with conditions. It can be flaming or smoldering. We know it's a complexity of the fires. You can play with the humidity levels and the water levels, and that gives you an idea. And of course, then you can mix things together. You can have different types of fuels, you can run them together. So that gives you the flexibility to understand a little bit how all these parameters affect chemistry of the emitted particles, and more importantly, what does it mean in terms of the bioactivity. So we published quite a few papers on the role of water, fuel, flaming versus whatever like conditions, complete combustion. And that enabled us to study this complexity and understand a little bit the impact. Not all fires are created equal, so they vary. And the LA fires, we talk about this was an endpoint interface. I show a few data there. When you start burning structures, cars, buildings, and plastics and other things, so the chemistry is completely different. And also, I mean, for the epidemiology, it's going to be even more complex because they use primarily the mass PM 2.5 metric. These particles that are emitted from wildfires are primarily nanoscale, so they're massless. They don't have much mass, so they won't really affect much the baseline in terms of micrograms per cubic meter, which is the usual metric that every studies are using. But that 1 or 2 micrograms per cubic meter that they're running in the baseline, especially far away from the wildfires, because of the new emerging chemical components of the PM and the bioactivity, that will change dramatically. Okay. And from our studies, we have a reference of all kinds of environmental nanoparticles. Actually, we're studying traffic-related nanoparticles. So we're collecting in airborne environments, traffic-related nanoparticles. We have the samples from wildfires, from the Canadian wildfires, or the ones that we simulated in the lab. I can tell you the wildfire nanoparticles are the most bioactive. And the wind fires are even more bioactive. Sorry for the long answer.
Question back there.
Yeah, I've always, having been in the field for a few years now, I'm a particle chemistry person as well. And so, you can make very complicated chemical measurements, I can make very complicated chemical measurements, but we still have a PM 2.5 and a PM 10 standard that doesn't consider composition. And intrinsically, we know that if I go walk by the beach and have some salt and compare that to wildland smoke, it's going to have a very different health effect. My question is, will we ever see? It was tough enough to get PM 2.5 as a criteria pollutant in the first place. Do you think this goes to either anyone out there really? Do you think we'll ever see something that includes composition or source based within the criteria pollutants? Well, that would be a great start, I actually think. But yeah, just in terms of actual chemistry.
I think the answer is not on the short term. I think that's pretty obvious. I think you go look at the PMISAs and look at the reviews and where they come out, for example, on ultrafines, and what might be needed. I think with this administration, the evidence bar is unattainable, whatever it might be. It doesn't exist, and so I think, going back to the debates around the PM 2.5 standard, I think when you start talking about national standards that cut across roughly every sector, there is this input, across the range of stakeholders, whether public through all the industrial sectors, becomes very, very powerful, and that played out in a way that's probably sort of quaint in 1997 versus what would happen in sort of the present landscape, and what science would sort of push us to say, well, we need PM 0.1 elemental, whatever. You know, I mean, I just I don't see us getting there in the short term. To the extent that epidemiological studies remain the primary driver, we won't have, it'll be very hard to generate strong evidence unless we monitor the array of indicators that might be relevant to changing the PM next.
In terms of, I guess, monitoring, though, one of the time that you're going to get it, right? Because if we don't monitor first, we won't know, and if we don't know, then we're never going to regulate it. So it's kind of, I guess, how do you break out of that cycle, potentially?
In a sense, that happened with the PM 2.5 standard, because much of the decision making was based on PM 10. But of course, you know, we knew a lot about particle deposition 30 years ago. And so I think it made sense. And monitoring capability had also advanced so that PM 2.5 could be measured. I mean, there was discussion then about PM 1, which also would perhaps make more sense in some urban environments. But so I think advancing monitoring technology would probably be needed as well in terms of the technical feasibility of monitoring and tracking attainment.
Need a microphone. Hi, I'm Takita Zaidama at the Atmospheric Sciences Department at Rosenstiel. This question is mainly directed at John. I was intrigued by your comment that organizing a community around a specific question that requires interdisciplinariness is a way to get people at the table. And I was thinking about workshops that have happened on wildfire and their diverse impacts. There have been several in the US that have been organized by federal agencies. I don't think the National Academy of Sciences has had one yet. It seems like we need to elevate that topic to the NAS level to really get people from different communities maybe to just bring it up to a higher level. And I was just wondering what your thoughts might be on that and what the process for that would look like.
It's a great question. I mean, clearly the National Academies is a useful venue for bringing people together, and the workshops and committee reports can be valuable. I mean, for those of you who don't know the landscape, the National Academies, those do workshops if somebody pays for them. And so it'd be important to find, you know, someone, and they can fund some things, and right now they're under siege, obviously. They have a climate and health initiative that having a workshop on wildfire smoke might fit into. I put together a workshop in August 2020 on airborne transmission of SARS-CoV-2. At the time, it was incredibly controversial, and I brought exactly what you proposed for wildfire smoke, you know, an interdisciplinary group together, and structured the workshop in a way that we could actually reach some sort of conclusions based on what the speakers said. We had 15,000 attendees virtually. And so I think you have a good suggestion. I think part of it would be to find someone to support a workshop, or, you know, going back to where we started around creating a research agenda, perhaps that would be useful as well. And I mean, again, I think it's possible to bring, you know, proposals to the academies. If it happens all the time, they're interested in taking on challenging topics. So I think if a group of funders could be assembled, I think it's one way to bring people together and put out a report that would have the imprimatur of the academies and hopefully carry some weight.
Yeah. Yeah, actually, that's a good question. I actually might post an entire panel. So, we've obviously a lot of interventions over the past 4 years. We've looked at how we can reduce exposures and thereby reduce harm. I'm actually interested in the perspective of each of the panelists. So what are the evidence-based harm reduction techniques, right? And so what have you all seen in your various experience? What exists to date? And what are the gaps in understanding new or novel interventions to reduce harm? And I'm going to actually pose to the group and then maybe kick it back about coke stove, my thoughts on coke stoves. I'll actually start with Holly. At the end.
As far as prescribed fire, it's actually, I was going to talk to you about this. We don't know much about it. Like our prescribed fire practitioners, they're big into what's called blue jean burning, like you could just go out and burn as you are. Not a lot of precautions are taken. You very rarely see any respiratory coverings. They'll just treat it like a normal day. So right now there's not a whole lot. No, they will wear Nomex and occasionally they will wear face coverings, but it's not the norm.
So again, my focus is on cancer, so it's a little bit different than everybody else on the panel. And we have been able to empirically demonstrate that decontamination post-live fire reduces the amount of exposure, both on the body and in the airway, to volatile organic compounds and heavy metals that are associated with this increased risk of cancer that we see in firefighters. And so most firefighters, not always in wildland fires, are wearing protective breathing apparatus. So again, part of that decon is to reduce exposure on the skin, which is the primary mode of, I don't want to say ingestion, it's the primary mode of exposure for these firefighters. And again, because we work so closely with firefighters, they're not so interested in the next peer-reviewed paper. They are in pain. People among their regiment are dying, and they want quick solutions, even if they're not fully empirically tested, to reduce unnecessary harm. And so this decon, which uses literally Dawn soap and a hose from the backyard, has made a significant difference in terms of the amount of exposure to soot, which carries the carcinogens post-fire.
I can share our experience with the lake fires, which is a major event. I mean, there are certain things that we know they work. For instance, filtration and using HEPA filters indoors. I mean, that's not rocket science, but in the case of, there are unpublished data that using HEPA filters indoors during the event can reduce your exposure to the drama. Translating data from the research into strategies, it takes time. So, for instance, right now we're studying, I mentioned in my presentation, the post-fire ash. This is after the event. So when we discovered that there are huge toxic compounds, including PFAS and other things in the ash that cover the whole LA metropolitan area, 9 billion metric tons of ash cover extensively the area. So usually the people, they go after the fire and they start sweeping their yards and without wearing protective equipment. So the emerging data from the research shows that there are some toxic compounds there. So, and I show one slide that shows that the inside can be re-aerosolized. So when you try to sweep it from the floor, for instance, indoors or outdoors, it will be resuspended up in the air. So that points that you need to wear, when you clean your house, you have to wear your mask, you have to take precautionary principle and losing your exposure. So I think we're learning as we go. But it's going to be more strategies, especially in the public domain, and we need to inform the public. And I think what Eric mentioned earlier, we need to engage the public and try to translate the findings from the research into strategies to protect the public health. So we'll get there.
So showed you the answer to your question 2 hours ago. In the accountability change, so what do? You change the source. Take lead out of gasoline. You change exposure. You stay indoors when pollution levels are high. you change the dose. You wear an N95. And then you can start to think about things systematically around how you actually reduce what gets into the lung. You clean the air, that reduces exposure. I mean, you can just go through it all. And I think in the case of wildfire smoke, it just gets more complicated. You can't change the source, okay? And you can possibly change the exposure based on behavior and other kinds of things, or cleaning the clothing, or changing the dose, wearing an N95 with points of high exposure. So think it through systematically, and then think about what works. The study of that Wayne Morgan did a long time ago on asthma, for example, where you do household level interventions. So I think you can think this through systematically, and you'll find points where we have interventions for different sources, and points where we don't.
I would say, like, low and middle country settings, it's complex, right? And so the air quality study was obviously negative. We know that indoor air pollution as a result of wood burning is also compounded by everything else related to poverty, right? And so it is a more complex issue of improving socioeconomic status to improve respiratory and lung health. There are a number of countries that are rolling out LPG stoves. So the Prime Minister of India has made it a mandate that all half this country will have LPG stoves by the end of his term. In Peru, they're actually giving subsidies for LPG distribution. And so I think we'll have more robust data in the very near future around LPG and health outcomes in the economy.
Just a quick comment. I mean, things can happen. So I've been going to Ethiopia for roughly 15 years on projects there. And I was there in December. I had been there for 3 or 4 years. The streets of Addis Ababa have been transformed. They've actually introduced policies to favor electric vehicles. They are redoing the roadways with a lot of social disruption. I won't go into all the complexities, but in 3 years there was noticeable change. Before, the taxation structure was such that everybody bought older vehicles and they stayed on the streets forever. And then, of course, if you know the landscape in Africa, so many of the high polluting diesel vehicles from Asia ended up there. And so there's a lot of black smoke as you went down the streets. And that's all changing. At least in Addis, I was surprised to see how quickly it changed.
Go, one more. 2 more. All right, we'll do 2 more questions real quick. Table here. Oh, yeah.
Incense burning all over the place. It's little oils, like scented oils all over the place. So we go around with this place going, shh, 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 shh. That's the smell. But those people do not really understand. They were doing more harm by doing all that than by they exposed for whatever went from the freeway through the walls, through the windows, through the cars. And there's a huge commercial value in all these candles, the scented candle. Home incense with the smell of pine and everything. And I think that people don't realize those things are not good for you. And I have not seen that many studies evaluating exposure and EPI studies from what the indoor environment from exposure to all those emissions, apart from cooking, because cooking has been studied for many years. But all those emissions, what would be incident need to the studies and bring this knowledge to the population and reduce all those emission sources.
I used to chair the advisory committee for the UCLA Particle Center. It's an interesting experience. Yeah, but do you need to study everything? I mean, I think it's sort of the common sense level of source management in the home, which there's a lot of discussion about. If you're burning incense or whatever, I mean, and actually there's been a lot of characterization of what burning incense does to indoor air. So you could start there and say, why not control it. I mean, I think the case of secondhand smoke, the epidemiological evidence on health effects, and particularly lung cancer effects on children, was really important to changing people's policies about how they wanted their home to be. But I mean, I think in terms of all the sort of cautionary advice, American Lung Association, EPA on schools, whatever, a lot of it is directed at sort of, I would say, common sense source control, which in a way would address your question. I mean, I appreciate what you're bringing up, but it's, I think, part of the broader question of thinking about the sources that you can actually manage within your home.
There's also emerging literature in terms of wildfires and what does it do to the indoor environment. There are all these semi-volatile organic compounds with part of the smoke that they penetrate indoors, and those are really tricky to remove because they partition between gas and particle, so they will stick on. So I can tell you more about it, but they can stick on the wall, on clothes, on everything. So after the fire, it will take a while. So ventilation will do it. So it's a question. This emerging literature which we need to inform the people and devise strategies how to deal with these semi-volatile organic compounds from and what you mentioned is true. I mean, the people, all these sources indoors, some of them that we introduced for whatever reason because of marketing and other things, and people are unaware of the consequences.
Actually, this is a posters you put up for signal. So, if there are remaining questions, first of all, I want to just thank the panelists for coming in. I think this is great. If you want to give any questions, and then someone please feel free to ask panelist questions after this session. Thanks, guys.
Thank you, everyone. And now it's the photo time. We're going to take a group photo. So please, everyone, move toward the end of the third floor. We're going to take the photo over there. Thank you.
Coastal aerosols and novel observational techniques. So I'll be moderating, and Sandra Gaston, for those of you who know, and then also Danny White will also be moderating as well. So we're going to have a diversity of talks that are going to focus on looking at impacts of harmful algal blooms, remote sensing observations near coasts, all the way to thinking about community engagement and how you get community stakeholders involved in some of these questions, as well as thinking about microplastics, as well as thinking about things like water pollution and how the water chemistry can actually impact air quality. So our first speaker is Andy Olt, who comes to us from the University of Michigan in the Department of Chemistry. So his background is, he focuses a lot on single particle techniques, characterizing them through a multitude of different methods. He's done a lot with spectroscopic techniques to look at fingerprints for different particle sources. And he's spent a lot of his career actually looking at harmful algal blooms that proliferate in the Great Lakes, which is also an issue that touches on us here in South Florida with some of the harmful algal blooms that we get, not just on the coast, but also some of the inland regions like Lake Luchui. So I'm going to be talking about some of his observations and also looking at whether what's happening in the water column can actually impact what's happening in the air. So with that, I'll turn it over to Andy. Thank you.
Thank you, Cassie, and to all the organizers for having me. It's been a wonderful workshop so far, so really want to appreciate that. I also just want to give the students a shout out going around for a lot of the posters. Those were fantastic, and I really enjoyed talking with many of you about the great work that you're doing, and so keep up the good work on that front. So I'm going to talk to you today about a couple of different things, and it's if my first true love is kind of secondary organic aerosol, then harmful algal blooms and freshwater aerosol are kind of our side project that we really love and have been having going on now for almost a decade. And we're going to get into a little bit about this. And so I like starting with this picture because for folks that maybe don't come to the Midwest all that often, up to Michigan, this is actually a picture from the Great Lakes. We do get waves. It's actually a thing that can happen. And as one of the largest, sort of areas of fresh water in the world, When these waves happen, it's not that different than what happens off the coast here, down in Miami, or down in Barbados at Cassie's site, in terms of the ability to produce aerosols. So I'm going to tell you a little bit of a story about that and related to some other work that we do as well. Before diving into all of the science, I want to make sure to thank the crew that is behind a lot of this work. The current folks on this project you're going to hear about today are Carly Barabasnik and Grace Young. But a lot of the work has been done over the years by Dr. Josh Shee, Dr. Nicole Olson, who's both graduated, as well as Nate May, who was a student in Carrier Grant Group that we worked really closely with, as well as, of course, we appreciate funding that helps us do these things. I will say this is a picture out of our lab on a really nice day in the summer. This is the picture I took out of our lab yesterday, so slightly different. So thank you also for the chance to escape Michigan in the middle of winter, although when it clears up after the snow finishes, you can get out to do some fun things like cross-country skiing. But it's a little nicer running around campus here this morning. So, what do we do in my lab? Cassie gave a little bit of an overview, but just to give you kind of the basics, we do a lot of secondary organic aerosol. We care about things like phase separations, acidity, viscosities, different organic reactions that are occurring. I'm going to talk a lot about our freshwater aerosol work, but we also try to get out into new areas. We've recently been getting into some microplastics, as well as getting out into the field to apply some of the spectroscopic methods that Cassie mentioned that we are really interested in as well. We always kind of have stuff going on the side as well. So we've recently built an optical tweezer, as well as we're doing a bunch of 3D printing of different types of aerosol size selectors and collectors, cyclones, things of that nature. So really kind of all over the place, but we're going to focus today on aerosols and both coastal, as Cassie had asked for, as well as harmful algal blooms. So just to give you an idea how we get out into the field, we do work with Cassie as well as other folks, including Carrie in Michigan, Judy Bustrick, who's at Wayne State, who we work on the harmful algal bloom stuff with, as well as Rachel O'Brien. And we are starting to get quite a wide swath of data from around the world in terms of different types of data sets. You know, for example, we've got microscopy samples that Phoebe and Sujan and my student Rebecca collected this last February. A sabbatical that was over in Australia last winter where we got a bunch of samples, as well as, you know, lots of stars over the US and Canada. So how do we go about looking at these particles? The work I've been doing ever since I started grad school, and even undergrad, was to look particle by particle at what's going on. So if we heard this morning from the public health folks about how we're going to look at, you know, a 5 microgram per meter cube change in PM 2.5, I want to know what that's made of. Where did it come from? What was the source? Is it primary? Is it secondary? How much of it is primary? How much of it is secondary? All of these different things that are going to tell us about where that particle came from. And to do so, we got to probe it in different ways. And so our sort of original way of doing this was with scanning electron microscopy, with energy dispersive spectroscopy, which tells us the elemental composition. But carbon being a really important element has a couple different forms. And so with that, we then get into things like Raman microscopy, optical photothermal infrared microscopy, which is very complementary to the Raman, as well as things that can tell us physical properties like atomic force microscopy or coupled methods. So we use these and we go to different places. This here is a picture from the field work we were doing at Kennewick Cape Grim on Tasmania. And for this, we were looking at some of the least polluted air on Earth coming off of the Southern Ocean and back trajectories. If you go back 48, 72 hours, you're going to head towards Antarctica type of thing. On the flip side, we also have been to Barbados with Cassie. As you can see, we're back up there in the corner to their wonderful site. You know, Anne, we started working with her back with her student Anne Barkley many years ago, and it looked at a number of different types of microscopy, such as from her student Haley, who graduated not too long ago, her work here using electron microscopy down in Barbados. So that's kind of the coastal angle. But I don't live near any oceans, and so if I want to think about what I'm going to work on in terms of bubble bursting, and I had done my postdoc, you know, largely out at Scripps and UCSD, we were looking at saltwater, but we don't have a lot of saltwater in Michigan. But we do have bubbles, and they do burst. And I showed you that wave on that first slide, and so when we think about it, when we started looking at this, really no one had looked at the composition of does something different come out of the Great Lakes that comes out of the oceans or any freshwater source. And in fact, most of the larger scale models just assume that gave the same properties as you would to saltwater, which is going to be quite different. And so as you think about these bubbles bursting, right, you think about the salts that might inhabit the interface here as this starts to fall apart. And as that bubble cavity collapses, you know, you're going to have the film falling apart, you're going to have the jet coming up the middle. And all of these are going to have different properties depending on how salty, how organic, or if I have a bloom around, how toxic my water is. And so if we kind of think about comparing the two sets of water that we might be looking at, if we're looking at traditional salt water, which we have over here on the right, this is concentration on the y-axis versus conductivity. Notice I didn't put salinity here because that doesn't actually officially apply to fresh water based on the ion ratios. But if we have conductivity here for a typical saltwater type solution, mostly sodium chloride, drop an order of magnitude to magnesium, drop another order of magnitude to get down to some of these other species, and eventually dissolved organic content in a normal area is going to be relatively low here as well. On the flip side, if you go over to the Great Lakes, you'll notice, right, we've dropped 3 orders of magnitude in terms of how much salt we have. So that's going to have some effects. You'll also notice that our dissolved organic carbon, it's right around the same concentration as a lot of the individual ions that are present. Why should we care? Well, that's going to impact as well how these bubbles either come up through the water column, how they coalesce, and eventually how they burst when they get to the surface. So there's actually some really cool old studies that people did to try to figure out the differences between saltwater and freshwater, where they actually would do these giant tipping trough experiments where you get a trough, fill it with water, dump it, and then they had a microphone under the surface, and so you would actually just collect the frequency that was released when the bubbles burst, and that would tell you what size the bubbles emit. So you can see frequency, the label looks like it got cropped, but the frequency is up top here, and then on the bottom we have the bubble radius. And what you'll notice is that saltwater has a pretty small radius for most of the bubbles. There's some that get up above a millimeter, and that's where that NT ratio is really important for the eventual formation of particles. But on the flip side, the freshwater has a mode that's quite large. You get quite big droplets. And so people have mostly focused on this kind of in the water context, but hadn't thought about it in terms of aerosols when we started getting involved with this. One really kind of simplistic study from a physical chemistry journal in 2008 is they actually took a series of salts and at different ionic strengths. They just had bubbles come out of a capillary. And you can see here that under high ionic strength conditions, the two bubbles, they can't merge. Whereas here, they merge really easily. And so this was actually quite in line with that prior slide, showing you that the lower your salt concentration, the bigger the bubbles you're going to end up getting. So how does that translate to the aerosols? Well, here we can see some synthetic seawater that we put into our little aerosol generator, which is described here in this paper. And you can see relatively small bubbles with your eye. And here you can see our synthetic freshwater, where we essentially, in the same way you buy instant ocean up here, we came up with an instant ocean for the freshwater conditions, and you can see that the bubbles do look bigger. And here's some Lake Michigan freshwater that we grabbed for comparison. But of course, it's very unquantitative to look at a picture and say, those bubbles bigger than those. So we can actually go to data. And we can see that our bubble concentration in the synthetic saltwater is about an order of magnitude higher than it is typically in freshwater. So you're getting a lot of bubbles. And if we look at the bubble size distribution, which I want to pause and make sure to thank Alexa Watson and Nate May, because they took a ton of pictures and came up with the image data scripts to be able to pull out exactly where the bubbles were. It was a ton of work. But what you can see is the synthetic freshwater does indeed have a lot of these smaller bubbles. Seawater, the synthetic, instant ocean essentially. Whereas the freshwater, it really drops off, and it's mostly these larger bubbles. And so when we first started doing this work, people said, oh, you're never going to get particles out of the lakes that matter because you have so little salt that the water will just evaporate and you'll be left with a tiny, tiny salt particle. But you get a balance between the fact that you have these really big bubbles that make bigger droplets that come out, even if they have less salts, and we can actually then start thinking about aerosol composition and size distributions. So if we go and look at that, here we can see the size distribution for traditional, kind of what you would expect if you went out to Scripps or somewhere else to look at the size distribution, out to Rosenstiel to look at sea spray aerosol. And you get about 110-ish. But when we go to look at this freshwater, you can see that for our synthetic one, which is in red, we have both an ultrafine mode as well as a mode out here at about 300 nanometers. So that was kind of surprising. We weren't expecting that. And then if we compare it to Lake Michigan water, we had one about 180, 200 nanometers and another ultrafine mode. So it wasn't just our formulation that was leading to this. We were actually seeing this bifurcation of the aerosol size distribution. And so we actually compared that to the one paper that when we started in this field, this was literally the only paper on this from Paul Shepson's group, Nate Slade, in GRL in 2010, had showed this size distribution, and they had showed, oh, there's this ultrafine mode we can't explain when we're flying over Lake Michigan. And so We contend that it's likely this. There are some debates. There could have been nucleation going on during this time. We don't know for sure. But we do know that this mode forms when we were observing it. And if we compare, you'll also actually notice that there is not just the ultrafine mode, but also this accumulation mode out of 200 or 300 nanometers. And that's actually this was in flying over a lake, so you have all the sources of the world. This is isolated to our little aerosol generator. And you can see that that very clearly does have an accumulation mode there. So, when we go to look at this, I don't normally show this slide, but I figured for this audience, since we're right on the coast of Miami, it might be of interest. We actually systematically decreased the salt concentration to see if in the salt water we could make those two modes appear as well. And as we go from sort of your traditional saltwater NaCl concentration down to closer to freshwater concentrations, you actually can bifurcate the mode there as well and get this smaller mode at about 100 nanometers in this case, as well as one out of 200 or 300 nanometers, which we were pretty surprised by. I thought was pretty cool actually, because this is in a chamber where you're not going to have anything leading to like a Hubble minimum type situation for cloud processing or something. So with this, we also can see that the number of particles we're generating as a function of salt concentration, it does vary. It varies, by a factor of 5 to 7, but it's not like it varies by orders of magnitude. We still get plenty of particles down here at these concentrations that we would see over the Great Lakes. So with this, I'm going to not tell this full story because there's a bit more that I want to get to today, but we did really characterize this freshwater aerosol in a nice way. And then we also did a study right out here on the shore of Lake Michigan. NOAA World puts out these great wave height maps. So you'll notice we're getting 2.5 meter waves. So, for those of us thinking in freedom units, we're thinking, 8 feet-ish or so. Decent sized waves here. And so we actually do see these calcium carbonate particles coming out of the Great Lakes, which is not at all what would be represented in a traditional source, assuming that it's sodium chloride. We also then showed that these don't just exist right on the beach, because here they actually got the car stuck in sand, trying to get right next to the lake to be able to sample these particles. While on a really windy day, it's not nice and 80 there. I think it was about 40 on this day in November, so they were a little grumpy when they got back to Ann Arbor. But we had that going on. But we also then looked, and this is a picture from that day with those waves. But then we also did some measurements at the University of Michigan's biological station, which if you ever talk to anyone from Michigan, they show you their hand like this. And so it's right up near the tip of my middle finger here is where the biological station is, about 15 miles south of the bridge that takes you to the Upper Peninsula, where we are able to we're about 40 kilometers or so from Lake Michigan, and we were able to show that particles with some PSCF, source contribution functions, we were able to show that the particles were coming from over Lake Michigan, and it was actually the background of about 5 to 10% of our PM2.5 was coming from this lake spray aerosol source. We also worked with Alison Steiner on her fantastic postdoc Anna Amiri Farahani, who was able to show that not only does the calcium carbonate that we've generated, it's really good at sucking up nitric acid, so it's actually a great sink for that. It does this reaction where it ends up emitting CO2 and water and leaving calcium nitrate behind. And it also then, once you've acidified it, it is a great sponge for soaking up whatever ammonia you might have hanging around. So that was another fun kind of story that I won't go into, but it was pretty good. The last one, kind of in this initial freshwater non-HABI portion of the talk, is that we actually, working with Paul Shepson's group, he flew both in cloud and above cloud, and we were actually able to show that the calcium carbonate from the water had gotten up into the clouds and was there when these clouds were forming over the Great Lakes. You can kind of see this bar chart comparison right here. And we actually talked to some folks at Oldemont and others who had done some flights over the Great Lakes 15 years before. And they said, oh yeah, we had this source of calcium in our clouds. We had no idea what it was, so we never published it. So it was actually kind of some nice confirmation that in fact we were seeing something that's been happening that people just hadn't been looking for. Okay, so we made some particles. They've gotten away from the coast. They can do some chemistry, and they can do some cloud stuff. That's all fun, but the title of the talk was harmful algal blooms. So let's switch gears a little bit towards that. If you don't pay that much attention to the freshwater HAB community, and you guys do have freshwater HABs down here in Florida as well, but for us, we really care about the western basin of Lake Erie, which is what you're looking at here. For context, if you're not used to it, that's the Detroit River. Detroit would be right about there. This is Toledo down here, and it's a pretty shallow lake. So every summer we get these harmful algal blooms, and they've been increasing in intensity. When I was a kid growing up in Ohio, everyone was like, oh, Lake Erie's back. It's, recovered after the EPA came along and cleaned things up from the 70s when Cleveland's river caught on fire. But by the time we got to about 2000 or so, we actually started getting these intense harmful algal blooms again. And it wasn't this time due to sewage or runoff from cities, but actually from a lot of nutrients coming from agriculture, such as phosphorus and nitrogen for fertilizers. And in doing so, these blooms have gotten more and more intense. It's not the same every year, it depends on how dry your spring is, how much runoff you have. But on the whole, if you compare where we were in the early 2000s to where we are today, it's much more, these blooms are much more intense. And you might say, okay, great, you got a green lake, who cares? But the reason this matters is that this little fellow microcystis here is able to produce a series of toxins, including microcystin, which is a hepatotoxin or liver toxin. Not great. And in fact, here you can see the Toledo municipal water intake, which is actually like kind of just off the coast here. We were out there on a boat last September. And this is what it looked like in around the time in 2014 when they started detecting microcystin, this toxin, at levels above drinking water safe concentrations in the municipal water supply for the city of Toledo, which is about 400,000 or 500,000 people. And they actually had to shut it off in this 4-county area for about 4 days. And you couldn't drink it. You also couldn't boil it. Because if you boil it, all you're going to do is pop these little fellows and release a whole lot of microcystin into your water supply. So it's not actually going to make it any better. So it was all bottled water, no showers, not great. So this really motivated a lot of work in the sort of Great Lakes region to try to figure out what's going on there. And we don't claim to be, special. This could happen anywhere, right? Where you have a lot of this runoff. So, what exactly is microcystin? For those that don't think about this all the time, microcystin here is this little ringed molecule. And it has a ton of different flavors or forms. We call them congeners, and you can have over 300 different congeners of microcystin have been detected in water. And what happens is, as hepatotoxins, they actually inhibit protein phosphatases. And we have a pathology slide on here. If anyone asks me a follow-up question on it, I'm not going to answer it because I don't know pathology. But I do know that this is bad. And this is not great for our livers. And we'll just leave it right there. So with that, we knew that this was an issue that we were interested in. So here's just a couple of pictures of these blooms across the Great Lakes. That's a fishing boat, for context, going out and taking people who, just like we heard about the quail hunters earlier today, here they're going out to go fishing on the Great Lakes to catch different types of large sport fish. Here we can see another one with some waves heading towards the coast of here. That's northern Ohio that you're looking at. Here's another one of the Maumee River, which is the part that comes down from Lake Erie and through Toledo. And here you can see another one with a couple of major roads going across. So this is every August, essentially. So it's not a really great situation that we have going on. People have been working on it, some successes, some failures, but there's a lot of effort going into this. And we were some of the first, I think we can say safely, to show that from these lakes, from the water from the Great Lakes area, we were able to make aerosol relatively efficiently. So we actually had to cover VSCT back in 2018 with Nate Haig's paper, where we compared water from a non-bloom condition over Lake Michigan to a lower bloom and a higher bloom condition over here in Lake Erie, and we were able to show a correlation between them. We then came back to that a couple years later because we showed that the aerosols come out of the water, then we showed they have organic stuff in it, this paper, and then we did a paper comparing two smaller lakes over by Muskegon in western Michigan, Mona Lake here and Muskegon Lake. And just to give you an idea, if you're looking at this plot that we have over here, these are July, September, June, and October for Mona and Muskegon Lakes. You can't actually see on this plot, so we have to do an inset to be able to see where the do not drink level is. And this is the level of microcystin that we were seeing in some of these waters. So again, not a great situation. And you'll see people out there like, jet skiing and swimming and all sorts of stuff, and I don't get it personally, but again, blue jean burns we heard about earlier. I guess people are gonna do what they're gonna do. But this is, not a great situation. And one of the interesting things that we saw in the study, this was led by Nicole, is that when we looked with triple quad mass spec and what was in the water, our two most common congeners were RR, two arginine groups on our microcystin, or LR, a leucine and an arginine. But when we looked at the aerosol, we didn't see the same congeners there. We actually saw a lot of LR, and we didn't see almost any RR. So the chemical composition of what was in the air was different than what was in the water. We actually could explain it by the KOW values of these different species, as MCLR has the highest KOW value. That means it loves being in the octanol the most. So it's going to partition to the surface of the water or the bubble, right as it bursts, and it's the one that's going to get spit up into the air. So when we see it, we were able to, so when we see this, clicker's going crazy, forgive me. One more time. Okay, so what we were able to see is that we were able to get these different congeners in the air versus the water. But again, this was in our lake spray generator back in the lab where we had brought the water. We wanted to go into the field to see if we could see this in the real world. And so we went to Grand Lake St. Mary's, which again, if you don't know your Midwest geography, is right here, about halfway up Ohio on the west side. And you can see here, this is the actual color of the lake behind it. I haven't done any photoshopping on the color. It's that green, and it's almost that green 9 months of the year. It's about 7 to 10 feet deep and about 20 miles across. So it is a perfect, and it's a very agricultural area. So it's a perfect place to build up nutrients. And as you can see, as the water hits the rocks here, some nice spray particles getting up into the air. So here, Nicole, as well as undergrad Isabel and Ja over here, again, no color changes, that's the color of the water. And here you can see us sampling next to the water back in 2019. And if we were to look here, this is a little small, I grabbed the text from a figure, so apologies you can't see that. But if we were to look for the drinking water advisory or the recreational advisory limit, this is over the course of a full season in 2011 and 2012. So you can see we're above that level almost all the time. And so we sampled at these 3 different places around here. Again, just a satellite image, it's that green, notice we're framed here. And we actually had a number of different ways we could make aerosols. We could have our traditional wave breaking. There were actually a lot of aeration systems. People don't like the green. So a bunch of the ridge strokes that have kind of their lake houses along the water will actually pay to put in bubbler to essentially clear up the bloom, and what they do is they're essentially just aerosolizing a whole bunch of stuff. Also, they'll bubble ozone through it to help, supposedly. Don't quite understand that one as well. Interesting chemistry. I mean, I find it interesting, but I wouldn't do that and then breathe it. And then we also had a spillway here, which you can see just depicted. And so when we got the water, brought it back to the lab, we looked at both the water and what was in the air. And so you can see the structures, they don't change a ton. This is our general generic structure with 2 different amino acids here in the X and Y position. Our R is arginine, and then we have a couple other small modifications, whether we have a methyl here or an H or what's going on up here as well. You can see we have an MC8 homotyrosine R as well as an LR here. When we looked in the water, it was pretty consistent across the lake which congener was the most common, and these are at our different locations that will have their different processes happening right next to it, essentially. And so with that, we're able to show that we had a lot of these species. But what was new for this study is that we actually collected air samples simultaneous to when we were collecting our water samples, and we were able to show, for example, by the spillway here, as well as along Prairie Creek, which is down here where I said those ridge strokes had all the bubblers, and we were able to see, hundreds of picogram concentrations of some of these hepatotoxins, which is, quite a concern. I did all that stuff. And we essentially just went around on a boat and sampled with a little 2.5 liter a minute filter pump, so with a cyclone in front of it, nothing fancy. And over here we sat, one of them we were on a boat and one we were right next to the lake. We actually saw this, we were collecting that a few slides ago. And so the key thing here, you'll notice the spillway had the highest concentration because they have a great aerosol generator, right? We don't have to take this back to the lab. They're doing this, this is about 20, 30 feet tall, so that's quite a drop. So you've got that going year-round under high water conditions. So, and here you can see sort of along the side of the lake, we also sampled those locations. We did some size-resolve sampling with a 5-stage high medium flow cascade impactor. And so this is going to, I'll just click all the way through. So this is 2.5 to 0.4, sorry, 250 to 440 nanometers up to 10 microns. And you'll notice that we essentially end up with a mode most likely around a little less than a micron, somewhere in the upper accumulation mode for where the toxins are in terms of what size particles their mass is concentrated in. And so we didn't just look at the aminopeptides, we also collected, oh, there you can see John collecting right by the side of the lake. But we also were starting to look not only at what was in the water, but also, so click, there we go. I'm actually going to skip this for time. Yikes. Okay. So the other thing we did is we were working, Lindsay Marr came out and her student Jin, and they actually collected on to look for RNA, and so they were able to actually see aerosolized gene copies at the same time, looking at this 16S rDNA. And we were able to detect that in the aerosol phase as well for some of the samples. So it wasn't just the toxin, there was, a whole bunch of the material getting aerosolized. We also were starting to look at other metabolites, not just microcystins. So here you can see amidopeptins. The structure is right here. It's a 5-member ring instead of a 6. But it has also some toxicity associated with it. And again, we can separate this into different congeners. And we see it both in the water and in the air. So we were able to see this. So here you can see the water samples and then the associated air samples. And we see different congeners, even between the different years, can change quite a bit as well as at the different locations. So we actually have this as part of a project. You can see Carly up here. We're working now with Jeremy Kadenko and Judy Westbrook over at Wayne State. Judy's the lead on our R01 project, where they're actually synthesizing some of these different metabolites, both microcystins and amidopeptins, to be able to see if these get into the aerosol phase. So again, we don't really do the synthesis, but we use what they have and then look at how well it gets aerosolized, because we're aerosol people at heart. The last thing I want to mention is that we do use new methods. I mentioned optical photothermal infrared spectroscopy earlier. I realize there's probably only a handful of folks in here that have ever really worked with that. And so the way this works is that if I want to get an IR spectrum, which is super useful for everything from microplastics, biological material, to dust, because you don't have to deal with the fluorescence you have with ramen, IR normally is limited to things that are 30 microns or so in size. Here, we actually get down to below a micron by using a green laser, a continuous wave green laser, just like I'm pointing at the screen. And essentially that's our probe laser, and then we will scan across with a tunable IR laser the whole mid-infrared, and we can get an IR spectrum out of it based on this relationship between pump and probe power. And so, for example, with everybody's favorite standard for aerosols, ammonium sulfate, we can see here ammonium sulfate quite well. The nice part is that simultaneously, we will also get inelastically scattered photons to give us Raman. So we get simultaneous IR and Raman at the same time. So we've done this for these things. This microcystins published this in JPIS 10A, and you can see that we're able to identify these particular types of particles, map them out, and we are part of a larger center that's looking at this in more detail now, including secondary aerosols. So that's the conclusions. I guess we're doing questions during the panel, so I will wrap up. And with that, I will turn it over to Cassie. Thank you so much for your attention.
Thank you so much. So our next speaker is going to be Lauren Zamora. She's actually an alumna from the University of Miami. She did her PhD at the Rosenstiel School between Dennis Hansel and also with Joe Prospero. And she is now at NASA, and she'll be talking to us about exploring aerosols and aerosol cloud interactions with NASA data. So we're very happy to have her, and we're excited to hear what you have to say. Thank you. Thank you so much. I'm really happy to be here. Oh, just to clarify, I'm actually with the University of Maryland, but I have an office at God. I'm a cooperative agreement scientist there. Right, okay, so thank you all for coming. So I'm going to talk a little bit about some of the NASA data that I work with, particularly in context of aerosols and aerosol cloud interactions. Okay, so we talked about this a lot, but of course, aerosols are harmful for our bodies in many ways. This is a picture from near Miami. They decreased visibility. This is from Reddit. It's a recent dust storm, I guess, this past year. They deposit nutrients on the ocean that affects the ecology there. And they change clouds and they change climate and weather. And so that's going to be part of what I talk about today. So aerosols, for example, they influence rainfall. They can delay or reduce rainfall. They can change rain intensity. They can alter global precipitation patterns. And they also affect storms around here. That's a concern. They can increase lightning, for example. They can affect storm lifetime. They can change heat and moisture profiles, and that in turn affects the clouds. And also, they warm and cool the planet. They can do that directly through absorbing or scattering light. And then they can also affect clouds, the cloud properties, and then the clouds will emit heat or they'll reflect light, and that changes the climate or the weather. So this is an image from Joe Prospero. He's a professor here. I worked with him, like Kathy said. This is, I think, the very first time that anyone saw dust from space. So it was 1970. And before then, they had thought dust was,
making these transit, but it hadn't ever been observed before. So satellites have been around for a really long time, and they've taught us a lot. Could you go ahead and press the, yeah, would you mind pressing, thank you. These are some of the currently active NASA satellites. So there's quite a few, and of course, ESA, that's the European Space Agency, has some. JAXA, that's the Japanese one. There's various, India has some, so these are just some of the ones that are up. We have a lot of aerosol observations, and I just put a list here. Is there a pointer? So this is a list of the ones that I'm aware of that have aerosol information, so there is a lot of data. Some of these aren't active anymore, and some of them are, of course, but there's just a wealth of information out there that people can use. So they work in different ways, so different types of satellites can tell you different types of things. So for example, some of them measure absorption at different wavelengths from, some of them look at reflectance, and so they go from, like, for example, the IR to UV and visible in between. They look at ratios between these wavelengths. They looked at reflectance spectra. They look at, if it's a LiDAR, they might look at backscatter and extinction and polarization. They might use polarization or reflectance at different angles to look at aerosols. And some of them don't just look down, some of them look across the, across the atmosphere towards the edge of the atmosphere, and then they can see what's going on with stratospheric aerosols. So they work in different ways and tell you different things. So one of the most well-known satellites is MODIS. This is an image of aerosol optical depth, AOD. And that just tells you some information about how much aerosols are in the full vertical column. And we have a long time series for this data set, and it's really nice because we get daily data, even sub-daily data, over the planet, including over oceans where it's typically hard to sample otherwise. It's important to know when you use these data that they do have some limitations. So for example, I mentioned it's a whole vertical column. You don't necessarily know where those aerosols are. So if you're interested in health or something, you're probably interested in aerosols near the surface, but they might be looking at aerosols 5 kilometers up or something. They can't observe aerosols very well below clouds or even above clouds for that matter, for this particular instrument. Humidity near the clouds can cause the aerosols to reflect differently, and that makes it harder to measure them. When there's low aerosol optical depth, it's a little bit harder. Near sunlit, that's when the sun is shining on the ocean and it gets bright. It's harder for them to see. And it can be challenging to separate the surface from the atmospheric signal. But these data can be really helpful for various purposes. So this is one study where they looked at, they used MODIS data, and then they combined it with machine learning, and were able to really well predict PM 2.5 when this is measured and this is estimated. They can do this on a daily scale, so it's really helpful for air quality prediction, for disaster response, for example, with volcanoes, industrial monitoring,
recording the Zoom,
and lots of other applications. And this, for example, is just if you're looking at the air quality observations here in Miami, they're pretty sparse. So that's another reason why, these satellite data are kind of nice. We also get vertical information throughout the profile of the atmosphere, depending on the instrument. So this is Calypso. It's looking at this really big Godzilla dust event that happened in 2020. It was the biggest dust event since another 17 years before or so. So you have optical depths of 5, which is really huge here in the Caribbean. And then the Calypso satellite told you where in the atmosphere it was. So luckily it wasn't, most of it wasn't near the surface, even though there was some here in the surface, but most of it was between 1 and 5 kilometers or so. So that's, you want to know that when you're thinking about health applications. So these aerosols tell you different things. They tell you AOD or backscatter. They tell you the shape and size of whether they're scattering or absorbing. They can tell you something about chemical composition. Most of them don't directly measure it, but they tell you with assumptions some information that can be helpful. This is kind of more cutting edge and a little bit less certain, but people are trying to start to use it to understand ice nucleating particles and CCN. They're even more assumptions. That's not real tested, but people are trying to push the edge of what we can do with these types of data. They also have limitations. Just generally speaking, clouds and humidity are usually a problem for almost all of the satellites. Composition is still pretty uncertain, particularly at low AOD. And there aren't so many validations for the aerosol type information. Okay. But another nice thing about the satellite data is we have long-term trends in data. So these are places on the planet where aerosols have statistically significantly increased or decreased in the past 20 years. So you can see here on the East Coast, we've had actually a big decrease in aerosols due to better pollution controls. But over in the Pacific Northwest, you have an increase, and that's because of all these biomass burning events over there. And then, this is pretty blurry, sorry about that. This is the United States here. You can't see, but. And then these red colors with hatches, that's the cloud droplet effective radius. And so, and this is for the same time period, this is like a change per decade. So while we've been losing or having less aerosols in this part of the United States, at the same time we've been having an increase in droplet size in clouds. And then actually it's not just where this is occurring, it's across the whole North Atlantic Ocean downwind. So they also tell us some composition information like I mentioned. So this, for example, is Calypso. You get profiles of, for example, backscatter and depolarization, and from that you can learn information about the aerosol subtypes. So here, blue is, for example, marine aerosols, and brown and yellow are dust aerosols. So now we have a new instrument called EarthCare. It's an ESA satellite, and they actually directly measure extinction, so we can do this even better now than we could recently. And then, of course, there are limitations with all the products. For these lighter data, the biggest one is that it has a really thin swath width. Like this is, it's like pencil thin, and so if you might have an interesting event over here, but no data because the swath width was so small. It loses signal with below optically thick clouds. So, for example, that's what's happening here. No data below this cloud. It's more challenging at low AOD, like all the satellites. Dilute layers can be missed. It's better at nighttime than during day. And sometimes it mixes up dust and ice, and I guess you can tell us more about dust near the surface. Apparently there's other issues he's telling me about. Oh, this is so blurry, sorry, I don't know what happened. This is another image of aerosol type from this is now from the MISER instrument. And it tells you just different mixes of classes of aerosols in a different way. It looks at aerosols from 9 different camera angles, and then it can say something about whether it's spherical or not, whether it's absorbing or not, and then they can subclassify that into lots of different groups of aerosol types. Oh my gosh, this is so bad. Sorry, can't see any of these. So this is these are single scattering albedos and size distributions from the AirNet sites. So these are what we use to validate a lot of the models, and we actually have one here at the University of Miami. This is a student of Paquita Zaidama's working on one. And so those are used to kind of help us make sure that what we're observing is correct. This is an example of use for, in this case it was the MISER satellite, looking at a volcano event in the Caribbean. And in this case, in St. Vincent, there was this big eruption, and then the volcano plume flew downstream over Barbados. And so people wanted to know, is this going to impact health for people? And part of what you need to know is how high that plume goes. That tells you, the air quality models use that information to see if people are safe or not. And it tells you where to go. So anyways, this product told us how high this plume went and where it spread. So other applications, these are very important for constraining climate models. So AeroCom, for example, is a big community effort to have a comparison between the satellite data and climate models. And in this case, for example, it was used to help model the direct radiative effect, which is important for example, for like IPCC when they make their reports. Oops. Would you mind playing that?
Sure.
So I'm not sure if the volume's gonna work here. I think I just have to narrate. So this is sort of a tutorial to show you how to use Worldview. This is a product that we use where you can easily look at data. So for example, in this case, we want to look at data from Florida. What's this happening? So, okay, so say we want to like zoom in there's this big smoke plume here that we might want to be looking at on this particular day. So you can click in and zoom in on any day, basically going back to the year 2000. You can click at different, you can click whether, say you want to look at the Arctic or subarctic. A lot of things you can do. You can click on add layers over there and look at different aerosol type, or different products like aerosol optical depth. In this case, we might want to look at aerosol optical depth years. That's one of the satellites, and pick. These are different algorithms you can use. We might just pick that first one. So you can add layers, and then you can see the smoke plume here in the data. And you can look at what the actual concentrations of the AOD are. And so here it's like closer to like 2, and if you go over here, it's lower. Some places you can't get the data because either there's clouds or the AOD is actually too high and it thinks it's a cloud. You can actually download the data and you get much more information, but this is like a quick visualization tool to look at what information there is from all the various products. Okay, so you can add another layer, for example. In this case, you might want to look at something like angstrom exponent, and so you can lots of different products you can pick from. In this case, it overlays it. You don't want to look at both, so you just unclick the other one, and then you can see where that smoke plume was. You have different angstrom exponents than at different locations. I had recorded this earlier with audio, so trying to make it here. So another cool thing you can do, this is a new feature, you can zoom in on a place like, for example, Florida, and you can kind of click over it and get statistics for that location. So say we want to look at this part of Florida, maybe we want to do it for like the full month of June. Some point I will click on, there we go. So we want to change it now from June 1st to, for example, June 30th. And then you can generate chart, and then it will calculate statistics for that location for you during that period of time. So it takes a couple minutes, but there you go. Now you're looking at angstrom exponent over that area during that time. So yeah, I think that's, we can, oh yeah, you can take photos, you can take images, and then download the image, but you can also, you can do it at different resolutions. You can get KMZs if you want to look at it in Google Earth. You can get TIFF if you want to do it in your own, like with with Python or something, or just images. So that's kind of a nice feature too. And I think we can move forward. Thank you. So aerosols impact clouds, I mentioned that earlier. These are some kind of like really visual examples of that, which is kind of cool. So here in the middle, these are ship tracks. You probably have seen these before. It's pretty clear that like a ship was here and it's impacting the clouds. Here's an interesting one from Florida where somebody did a loop-de-loop here. So it's like clearly not a natural signal there. This is an interesting case there's a copper plant in Canada, and then downwind of that copper plant, the aerosols nucleated the clouds and caused them to glaciate and then precipitate. So these clouds are really different from the clouds downwind of that plant. And then again, here's Florida, and this is where aircraft have passed through this cloud deck, and again caused them to glaciate, and then that causes these really interesting cloud patterns. So aerosols impact clouds. Usually, so these are easy to see. Usually, it's much harder to see. And this is in part because the effects depend on weather conditions, and in part because aerosols and meteorology vary together. So one's changing at the same time as the other, so it's really hard to distinguish the two. And these are some of the biggest uncertainties in climate science. So that this is the uncertainty from ineffective radiative forcing from aerosol cloud interactions. And this has a lot of repercussions for lots of things. Like our uncertainty in pretty climate impacts our climate risk assessments for insurance, for investments, for water resources. There was recently, Paris Accord being in the news. Even here in Florida, there was recently this bill where all airports in the state have to report any geoengineering and weather modification issues. So this is also related to aerosol cloud interactions. So I know this is mostly an aerosol group, but I'll just talk briefly about what cloud data are available. There are lots of other instruments. This is probably not comprehensive. These are just some that I know off the top of my head. We measure lots of different things like cloud fraction, cloud albedo, radiative effects, precipitation, phase and rate, cloud top height, temperature, cloud optical thickness. liquid ice and water path, vertical velocity in the cloud, droplet size and concentration, particle phase. So as with aerosols, there are challenges you need to know about if you use those data. The passive imagery can best observe the top part of the cloud, but sometimes you want to know what's going on in the full level of the cloud, so that's a challenge. And so you can use radar, like active sensors like radar or LiDAR, but the radar can't detect near the surface. And sometimes you care about clouds right near the surface, so that's a limitation. The LiDAR itself gets attenuated in the thicker clouds, and then thin clouds can be missed. So these are all some limitations of those data. So here's another visual. This is another way you can download data. This works for both clouds and aerosols. In this case, I'm going to show you. This is Giovanni. You can go to that website up there. In this case, I'll show you a cloud case. So you can look at lots of different properties and then visualize them. So in this case, we'll look at type average map, but you can look at like cross sections, scatter plots, histograms, and so forth. And so we're going to click 2005 in June, going back to that same month again that we saw before. In this case, just like a week, because it takes them like a while for the data to load. So you have to select your region, that's Florida again right there. And then you can select what you want to look at. So for example, observations, you might want to look at measurements instead of models, but you can look at model products too. Maybe cloud properties. And then you can look at the different instruments you want to look at. So we might look at MODIS again for this time. And then on the left here, you can see all the different properties that you can get data for. And what satellite product they come from, and then what temporal resolution. So maybe we don't want to look at monthly, maybe we want to look at daily. So you can go down to daily, and then you can look at the different time periods where those data are available. So say we want to look at liquid cloud droplet effective radius right there, so click on that. And then you can say plot the data, and then it has to take a little time to load. But it's a really nice feature because it's a way to really quickly visualize the satellite data instead of having to download everything and like come up with code to plot it. You can just quickly visualize what data are there. What this is plotting is going to be a pretty coarse resolution. If you want to go down to direct data, you can get it at much finer resolution than that. But this is just for quick visualization. So you have options. You can download the data in various formats like NetCDF. You can just get an image. You can get a KMC again. So this just makes it really easy to work with the data and see what information is available. You can plot it at different scales. I think we can move on, but this is just to give you guys an idea of what quick ways to work with data. Okay, so I mentioned that it's challenging to kind of separate aerosol effects from cloud effects, and this is just a visualization of like why that is. So say you have like this big dust plume blowing off of the Sahara. This air mass is hot, it's dry, it's windy, and it's stable, and it happens to be dusty too. If you go over here, it's the opposite. It's cooler, it's moister, it's less stable, and in this case, it's cloudier. And the question is it cloudier because there's less aerosols, or is it cloudier because it's moister? So the challenge is to distinguish these two things. Oh, so blurry, sorry. So what we did in this paper, we looked at deep convective clouds over the North Atlantic. This is a cool image from the International Space Station. And you can see these deep convective clouds in a storm event. So we used multi-year satellite data for CloudSat and AIRS, two different products. And then we used a real analysis product from ERA2 because of forget that part, the high cloud cover already. This is for the south. That was old text, sorry. This is actually in the subtropical North Atlantic. So we tested which aerosol meteorological parameters would best predict the differences between when there's clouds and no clouds. Clouds with low dust levels. And the key is that we have to do it in similar meteorological conditions, so it's a fair comparison. So we basically just bin by those data and then compare within those small subsets of meteorology. And then when we did that, we looked at which factors best predict the aerosols. And sorry, so blurry, but these black bars are meteorological variables, and these color bars are different aerosol parameters. For example, like dust, sulfate. Here in blue is sea spray, and in green is DMS, which isn't an aerosol, but it's a proxy we use for new particle formation. And then we looked at like which of these factors best explain the differences in these similar meteorological conditions. So we thought that dust was going to be kind of affecting these deep convective clouds, but it turned out that actually it was marine particles associated with the dust. And so what we think was happening is that these small particles were CCN in these deep convective clouds. There's a lot of vertical motion in these clouds, so they can nucleate. But when dust was there, they would probably coagulate. We hypothesized they would coagulate onto the dust. The larger particles, those would fall out, and then they're no longer in the system to act as CCN. So that's one method of doing things. There's also now like a lot of really interesting machine learning methods to kind of do something similar to disentangle the meteorology from aerosols. And this is just one example from a recent paper that I thought was pretty cool, where they took data at in the Midwest, and they looked at liquid water path and cloud droplet number. Didn't see much patterns, and then they applied their machine learning analysis and were able to kind of find what they believe should be and the actual signal minus the meteorology. So it'd be interesting to read. It's hard to explain, but you can read the paper and read the details. So a lot of things are now possible that weren't possible before. So lastly, I just want to talk about some kind of information we need in this field going forward. So this is going to be mostly focused actually on Arctic aerosols, which is where my background mostly is these days. But a lot of it's applicable to other locations as well. So secondary ice production processes are a really big uncertainty. Those are basically those don't actually depend on aerosols, but they're hard to, again, distinguish from aerosol processes. What we need these days are also continuous long-term observations of cloud active particles, like CCNs and IMPs. So there's earlier sessions talking about miniaturized instruments. That's the kind of thing that we could really use, observations of those at different altitudes, for example. It would also be helpful to have this information at long-term observatories, coordinating especially with LiDAR data, because then we could try to get more context historically for understanding the data. Getting more information on cloud phase partitioning from remote sensing, better integrating the lab and the communities is something that we still need to do. Improving satellite resolution and so forth. So like that. And then I think this is your last slide. Just another for those of you in the field who are interested in kind of areas that might be worth going into in the future. So AI ML is going to be really useful. It's mostly untapped in places like the Arctic where you don't have a lot of data. There are some limitations with applying those methods, so we need the data in addition to these methods. Having more regular profiling with uncrewed aerial systems and tethered balloon systems would be really great. And then coordinating those with field sites, optimizing how we design our campaigns to include that data, be really good. People are kind of excited about this new EarthCare satellite because it has Doppler radar, which wasn't available. So now we know more about vertical velocity in the atmosphere. So studying how that affects aerosol processes and clouds would be really interesting. And I think I kind of alluded to this before, but people are trying to get better estimates of CCN and IMP from LiDAR. So that's something that we need more testing and development. So this is my last slide, just takeaways. There's a lot of data that are available for aerosol and cloud research. It's all free. They offer wide spatiotemporal information and vertical information, and even some information on particle composition. And just if you use them, it's important to be aware of their limitations. Thanks.
You during the panel, and I'm sure there'll also probably be some requests from you for some of your slides, especially the tutorials. That was very nice to see. So we're actually going to have a little bit of a change in our program. So instead of Helena going next, we've asked Sung Yoon Jung to go next, and she will be talking about methodology to measure and quantify microplastics, which is another emerging concern in coastal regions. So take it away to Sung Yoon. Thank you very much.
Thank you. Thank you so much for
Hello everyone, my name is Sung Yoon Jung from University of Florida,
And today I will talk about development of standardized methods to extract and digest microplastics in environmental samples. My group has been working on two different research topics. One is the method development, and the other one is collection and analysis of microplastics from different environmental samples. And here, this one is one of our research. And this result was published in Aerosols on Analytical Chemistry last year, and I will discuss about this one in detail today. Yeah, so whenever I start to talk about microplastic, I always start this sentence. Plastics are everywhere in our life and extensively used in packaging, textile, and household goods. And you can find the plastic really everywhere, even this room, when I can see the water bottles everywhere, and then I can see we are wearing the clothes as well. So, since the 1950s, global plastic production and consumption have risen exponentially, as you can see from here, and significantly increasing plastic waste. And the majority of plastic waste was disposed in landfill or through dumping with only 9% recycled. So in the environment, these plastic waste generally undergo various degradation process because of different environmental factors such as UV exposure to form smaller particles. Among those smaller plastic particles, microplastics are considered to have their sizes above 1 micrometer with an upper threshold of 1 millimeter or 5 millimeter. And recently, many studies have detected and reported the presence of microplastics in different environmental media, including everywhere. So soil, water, air, really everywhere. And once the samples are collected, researchers generally use a multi-step treatment process, including extraction and digestion, which I will explain more in detail in the later slide. But using these different techniques, there are research gaps, which is insufficient information available about efficiency and proficiency of existing methods without changing the physical chemical properties of microplastics. And reduce the physical chemical property alteration is really important, and I will explain the reasoning in the later slide as well. So with this research gap, we defined 4 different objectives. First, we evaluate 72 extraction and digestion methods applied to standard and commercial plastics. Second, we assess the physical chemical properties of plastics before and after treatment to see the changes. Third, we compare existing methods with varying exposure duration, chemical agencies, and the temperatures. Lastly, we address the challenges presented by the potential degradation or loss of materials during the process. So throughout our research, we wanted to identify if there are changes under different treatment methods, and then what can be the proper method to use for our study. So here from this slide, I will explain the detailed methodology we used. So in this study, we tested the main categories of standard and commercial plastics commonly reported in the article, and we used different extraction and digestion methods in recent atmospheric research articles. So we utilized 4 different plastics, which are PVC, PET, PS, and PP, and their sources and images are available in this figure. And once we have these materials, we use 2 different methods, which are the extraction and digestion. Extraction uses generally density separation method to separate light plastics. So plastics are generally lighter than environmental matrices. So if we are using dense chemicals like that, then we can separate the plastics from environmental medium. And many people use different chemicals and also different duration. So we try to understand short, minimum, and maximum duration with different chemicals and to understand the changes in physical chemical properties of microplastics. And digestion is used to remove organic matter as much as possible. And people use different chemicals and mainly use H2O2. And people use different temperatures and also duration as well. So like extraction, we use maximum and minimum temperature and duration to see the changes.
So once the samples are prepared, researchers generally use different analytic instruments. And in our study, we use pyrolysis gas chromatography, mass spectrometry, PyGCMS, to identify and quantify microplastics. It's not compared, but there are always advantages and disadvantages of the instrument. And in our case, we choose PyGCMS because this machine does not have size limitation, and it has high sensitivity to measure tiny amount of microplastics. So once we run the sample, PyGCMS can create pyrolysis gas product, which can be transformed to GCMS to be separated and identified, and it can generate this kind of the total ion chromatography. So the first figure shows total ion chromatography representing polystyrene PS, and the second one shows the total ion chromatography representing for polyethylene PE. As we can see from here, those two total ion chromatographies are different. And people use these different total ion chromatography, and they identify different indicator ions to identify polymer type, and also we can normalize these indicator ion for the quantification. So in our case, based on our literature review and our experience, we identify those 4 different indicator ions for our testing material, and we investigated any changes in normalized area of this indicator ion to see the changes. So from this slide, I will show you the representative result we obtained. As I mentioned, we tested 4 different plastic types, and PET was one of them. PET has been widely used to make, for example, water bottle, and we found different variations in changes of microplastic properties. So x-axis show the chemical agent and also different temperature under different variations, duration. On the y-axis show the normalized indicator ion peak area. So we obtained this one by obtaining the area, integrated area of indicator ion divided by the mass of the original material. So the black bar is the normalized indicator ion P of original material, and the others are after the treatment. For example, if the peak of after the treatment shows similar to that of original, which means there's not much changes, and which indicates there's not much significant influence by the treatment method. And we found various changes depending on the method, including chemical duration and the temperature. And we would like to highlight some results. So first, we found that the extended exposure, like Ki and zinc fluoride after 27 hours, shows significant decrease in the indicator ion, the normalized area, which indicates that these duration may lead to degradation of certain chemical structure, in our case indicator ion. And the right figure shows that this indicator ion peak was completely decreased and disappeared after 72 hours. And we found that compared to the extraction method, digestion method shows relatively less influence on the normalized area. And in the case of PVC, which is also widely used for different products, and one example can be PVC tubing, and we can see the tubing everywhere. And PVC shows also different results compared to PET. So one distinct result can be Ki treatment. So Ki treatment does not show significant changes compared to the PET. Earlier slide shows clear and distinct changes with under Ki treatment in the case of PET, but this case does not show significant changes. And we also found that NaCl at longer extended duration showed a significant decrease in normalized indicator peak areas, indicating a substantial effect on the chemical structure. And we also found the negative impact of extended exposure of PVC to the digestion chemicals, and we also saw the color changes from these microscopic images. And in the case of PS is also widely used and also one of the dominant polymer type reported in the environment, and which is widely used for different products, I can say styrofoam. And this one also shows different varied changes under different treatment. And we found that relatively minor effect of the extraction treatment was observed. However, we found significant changes due to digestion treatment, and which indicates that PS is greatly affected by the digesting technique, and we can also see the changes from the microscopic images. Lastly, PP also shows different trend compared to the previous plastic types, and by the way, PP is also widely used and also mainly reported as one of dominant polymer type in the environment. And you can find this material from straw or water bottle can. And compared to other materials, PP shows relatively smaller changes, which indicates that PP has high resistivity to the treatment method. And in addition, it also shows negligible changes under extended duration as well. So with our result, we concluded that depending on polymer type, there are different changes in their physicochemical properties under different treatment methods. So based on our result, we thought different strategies for further direction. Of course, there's no single method can be defined as the optimal method. Rather, we would like to say a less aggressive protocol may be beneficial for studies that screen a broad range of plastics. And based on our result, most extraction methods resulted in generally mild changes in the physicochemical properties of microplastics, except for NaCl with PET and PVC, and zinc fluoride and Ki with PET, especially for extended duration. Compared to extraction method, digestion method shows a more distinct impact on microplastic, but compared to Fenton's reagent, we found H2O2 at lower temperature may be better recommended for study. Even though we studied and we concluded the findings, we had limitations as well. First, we did not examine all commercially available plastics. We only tested 4 different types, even though they are commonly reported as the dominant plastic types. Second, we tested relatively large microplastic, which are around 1 millimeter. And we don't know about nanoplastic or smaller microplastic. They may have different traits. Third, zinc chloride and the potassium iodide were found to be the most effective agent, but they were found to be toxic to organisms. It means we may need to find something else. Lastly, a potential limitation probably related to the selection of indicator ions and the use of peak intensities. In this study, we used certain indicator ions and intensities, but other people also found other types of indicator ions as well. So if we can find, or if we investigate different types of indicator ions, may be beneficial. So our study concluded that extraction and digestion methods shows differing impact on individual plastics. We emphasize the importance of selecting proper extraction and digestion techniques to minimize the changes of microplastics properties. And lastly, optimized method can improve the precision and reliability of microplastic concentration in the environment. Thank you so much. And since I have to leave early, I will be really happy to answer any questions through the email. Thank you.
Thank you very much, Sunyoung. So our next speaker is going to be Milo Mejia. He is from Catalyst. So this is actually going to be a really nice talk, segueing from our earlier session talking about community engagement and how community stakeholders can shape science. And so that is going to be what Milo will talk about. So thank you.
Thank you. Hi, everyone. So, yes, my name is Camilo Miguel. I'm the director, actually senior director of policy and advocacy at Catalyst Miami. And, well, you can see some of the things we do there. So Catalyst Miami was actually founded by our mayor, Mayor Levin Cava, right after Welfare Reform Act was implemented, leaving serious gaps, especially for low-income women of color. And so she created the Human Services Coalition that led to the foundation of Catalyst Miami to provide services, but also to bring people into advocacy so that they could participate in advocating for policies that had a direct impact in their lives. So when we talk about policy and advocacy with community members, we are basically not necessarily too focused on policy. In this particular case, we're not going to be too focused on science either. What I tell to people when we first start like a leadership program or a workshop or something like that is that if they have children at home and there's a rule not to eat cookies before dinner, but then the child comes to the mom and says, well, what about if it's a sugar-free cookie and I already did my homework? And the mom says yes, or the dad, well, that's policy, then that's a policy change, and that's advocacy, right? It exists in our lives. It's not that complicated. But they make it complicated on purpose because they don't want regular people to be there at the chamber, you know, when policies are being discussed and there's serious money on the line, but there's also serious impact in the lives of people on the line. So people should have the information that they need to be able to advocate for themselves and their communities. And so these are some of the areas of work. Clearly, I'm here because of climate and environmental justice, but we also do economic mobility and resilience, policy and advocacy and leadership development, as I just explained, and then community-driven research and action, which is basically what I'm going to be talking about here. Okay, so this is more or less what the county is doing. This is what the county does when it comes to monitoring air quality. Whenever a new business comes to town, they are the ones who issue the permits to ensure that these entities comply with local, state, and federal standards for air quality. They monitor and they ensure compliance. And so there's a whole department in the county that people can go to and say, hey, my air is polluted, but people don't know that. And we don't really have the language to talk about air pollution or the means to understand it. And so this is the criteria that they use to understand the quality of our air. This is not common language in our community. People don't really know. People know that they have asthma, that they have respiratory conditions and things like that, but they're not entirely sure what's causing that. What they are sure of is that it's not good to have a highway next to where you live, or to have a power plant or a motor pool, which often happens, you know, through zoning laws in low-income communities. And so with this, the sensitive populations, children, the elderly, people with asthma are most affected. Those are some of the main groups that we work with. And so the initiative that we're a part of is basically funded by the EPA. It ends this year. We're probably gonna have to revise that because we started a little bit late. The county is the lead partner, but then also we're also partnering with FIU and UM. And the goal is to expand and strengthen partnerships to co-design a clean air community partnership program that informs action. And our part is basically to engage residents in monitoring and decision-making to address pollution and cumulative environmental stresses. And so these are some of the goals and objectives. Basically, so I'll share with you, I don't know how much time I have left, maybe 5 minutes.
Close to 15.
Close to 15, okay. So some of the things that we are doing, we want to make sure that people are part of the conversation. So we're running a survey campaign where we're asking people about pollutants in a way that they understand it. Do you live near a highway? Are there a lot of businesses? What kind of businesses are in your community? And how is that impacting you? What are some of the health consequences that you see in your home, in your community? What are you hearing from people out there? Is it asthma? Is it higher incidence of cancer, as we have seen in areas where they have, for instance, built like an incinerator? You know, that's one of the most infamous historical references that we have, and they're about to build another one, bigger one. But so we're trying to understand air quality from a community perspective so that we can connect the science to the lived experiences of community members who don't have the time or don't have the expertise to be fully involved the way that maybe most of you in this room would be. But that at the same time, they have the tools that they need to understand the basic science and to understand how pollution impacts our lives. And then also to take matters in their hands, to affect change. Prior to this project, I worked, little side hustle, as a coach for EPA community change grants. And those community change grants, they were like gigantic community benefits agreements paid for by the EPA from 2 million to 20 million. And one of the things that they were looking for in their criteria was to create co-governance, to right historical wrongs. And you'd be surprised at the number of applicants who had air monitoring stations as part of the way that they measure not only environmental pollutants, but also the degree to which people could be a part of the solution. How? Well, by installing air monitoring stations in people's homes, in people's communities, and then teaching them about the different pollutants and how to read the monitors, and then how to use that for advocacy to create a co-governance model so that when the next developer comes to the county or the city and they start discussing the new motor pool that they want to build, for instance, for the shuttle, for my University of Miami shuttle, or like a city shuttle or a county shuttle, that they don't put it in the West Grove or Overtown or some community where historically we have seen this type of projects being built. And so by having this, we're able to integrate monitoring data into community networks and advisory groups. That means that people now have the access to go to a particular place or even their home where they can read the quality of the air and how that air is affected not only by specific events like the Saharan sand dust that comes to us every year, or the burning of an incinerator or any other event that may take place, but also their day-to-day air quality, right? And by them having that tool that connects them to scientific data that they can use to connect to their personal lived experiences to bring to a county commission meeting and say, hey, we don't really want this here. And we don't want it here because this is the impact that's going to have in our lives. And this is the data that connects to that, and this is how we know. And so it's a way to build trust and understanding between residents, scientists, and government. But it goes beyond that because it puts the science in the hands of the people, regular people, day-to-day people, to be able to go to the mayor and to go to the commissioner and say, hey, here's the data, and here's data that we have been a part of collecting and analyzing and understanding, and this is why we don't want that motor pool to be built in our community. This is why we don't want that bridge, we don't want that, whatever the case may be. And so what we're doing is we're having sort of focus groups with community members, and we discuss the different questions of the survey. And the idea is not only to collect information directly from community members, but also to identify community members who might be interested in being a bigger part of the project by allowing the project to install an air quality monitoring station in their home, and to teach them and to train them and give them the capacity that they need to be able to involve their fellow community residents, their relatives, their friends, etc. And then also work alongside the county. The county not only has this department that does the monitoring and compliance, but they also have air quality monitoring stations throughout the county. We want to make sure that we are getting the proper reading. If you don't have air monitoring stations in a place where they're building a lot of commercial sites or industrial sites, then we're not going to get that data is not going to have the weight that it should have in the overall picture. So we want to make sure that we go to communities where this has happened historically and install those monitors and then train people and then create a sort of movement and a network that connects those community members, not only with scientists, but also with elected officials, so that together we can right this historical wrong. So I think I explained that's one. Well, I think that's it. Well, and then I'll send you the, this is the contact information if you want to be involved. And then I'll send Jan the link to the survey if you want to take it. And if you have any questions, let me know. Of course.
Yeah, so that last slide you saw, that's actually a project that's spearheaded between University of Miami Catalyst and FIU, looking at air quality in different communities in Miami. So that's something that this group has been a part of. So if you have some questions about community engagement in science, it's a good group to ask. So our last speaker is Helena Solo-Gabriel. She is faculty here in our department of, or in our College of Engineering. And she's done a lot of work looking at the movement of contaminants and pathogens in the water, and she'll be talking about how that can actually impact our air quality. So Helena, thank you very much for joining us. Thanks for this opportunity. I really appreciate the opportunity to describe some of the research. And as Cassie mentioned, I tend to focus on the water and the sand. But I would like to talk a little bit about the aerosol work we've done and what we're doing now. So there's two projects. We've done many projects in this area. My favorite area to work is the intertidal zone, the zone between the sand and the water, where the water goes back and forth. And the two projects I wanted to describe first was our study that originated from the water. We wanted to look at the surface microlayer and see whether or not aerosols were coming off and understanding those aerosols. And then also a second project which gave us more insights as to what is going on in this intertidal zone in terms of the seaweed and sargassum. To give a little bit of background, why do I look at the water? Why am I interested in the water? Our beaches are maintained open and closed depending on the levels of fecal indicator bacteria. It indicates the presence of sewage at our beaches. And the way we measure this is using a microbe called, a group of microbes called enterococcus. And the way we sample that is we take 100 milliliters of water, we put it through a filter, we then put that filter on an autoclave, we incubate it, and then we count the colonies in colony-forming units. And if you have more than 70 colony-forming units, that beach is closed. This beach would remain open. Why are we concerned? What is going on? Well, in Miami-Dade County, these are the advisories. Since 2016, the numbers are going up exponentially. We want to know why. Doesn't matter if you are averaging the enterococci or if you're looking at percent exceedances, the numbers are going up. And we want to understand better so we can keep our beaches open. So this project, actually, we got a lot of help from Cassie on this project. A lot of the work we did, and one of the areas I focus on is the supra-tidal zone, that white zone right above the high tide line. The reason I'm so fascinated by that area is because that's where all the microbes accumulate. So we know that the microbes accumulate there, but we don't know why. And in talking to my oceanography colleagues at Erasmus, they told us the upper edge of the wave, and then there's Stokes Drift, and the microbes accumulate there. Yeah, I can see some accumulation, but we have a lot there. So, what else is going on? What else, why do we have so much bacteria up in the supratidal zone where the white sand is? So we hypothesized that perhaps maybe aerosols had something to do with it. So looking at the cartoon at the top, again, a lot of the sampling focused on the water. We have a transit, we start at the sediments, we have our aerosols, 3 samples collected there. Then we collect water in ankle-deep water, so we go and get some ankle-deep water, then we go a little bit farther to get knee-deep water, and also the surface microlayer. Then go a little deeper, we go in waist-deep water, and we collect at all different depths, and also the surface microlayer. We did this sampling event 15 times, and we were lucky that we captured a couple of rare events, including a sewer spill and king towels. At each site, we analyzed those samples by culture, for general marine bacteria. We analyzed for our indicator Mycobacterium pterococcus, and we also processed samples by sequencing. This is the setup that we had. We had 2 impactors and 1 surface filter. The impactors had agar in them, one specific for pterococcus, the other specific for general marine bacteria. This is what our plates look like. If you compare the water on the right, the white filters, to the aerosol samples on the left, the contrast, the visibility of the colonies is so much easier on the water samples. It's hard to see yellow colonies on yellow plates. Also, there's a lot of dimpling because of the way the impactor works. And in addition to that, the numbers are very low. We had no detections in the aerosols for enterococci, and then we had no detections for general marine bacteria, in that the detection for general marine bacteria in the aerosol tended to correlate with the levels in the surface microlayer. But continuing on, we had that 3rd filter, which was used for sequencing. And with sequencing, what we found was comparing the aerosol to the other samples, the sediment sample had the most diversity, the most different types of genus in the microbial community. The surface microlayer was second in line. Even though the levels of bacteria tended to be a little lower in the surface microlayer, they were more diverse, and similarly the aerosol also tended to be diverse. Now the aerosol had really low levels of bacteria, genetic material that we could capture, and yet it was very diverse. In terms of the multidimensional scaling analysis, which tells us a little bit more about the differences in the microbial communities, the organisms were different, the communities at the genus level were different between the aerosol, the sediment, and also in the water. The microlayers were more similar to the aerosol and sediment, and the bulk water, which is given by the light green, was more unique in itself. Looking at the microorganisms in more detail, this is a bubble plot, and you can see the aerosol samples in the middle there. There's a lot of white in there, but there's a lot of big circles as well. We have fewer organisms, but there's more diversity, and they tend to vary. So in the sewer spill, they tend to have 3 forms of bacteria that had high abundances, including forms that or genus that were pathogenic. Also very interesting, in the aerosol, we found high abundances of Staphylococcus, which we've been studying extensively in the water and the sand. And it was interesting to see that it accumulates, it enriches within the aerosol phase. And then also just to emphasize, only half of our samples were suitable for sequencing analysis because there was just not enough material to capture. We ran those systems for 3 hours, and it pumped more than 2,000 liters of air, and yet half of the samples failed. So it takes a lot of air in order to process the samples and to get enough material for sequencing, for instance. So for project 1, what we found is that there were distinct communities between the water, aerosol, and sediment, and we also found pathogenic bacteria in the aerosol, including Staphylococcus, Streptococcus, and Vibrio. That brings us to project number 2. And this project number 2 occurred by coincidence during the COVID shutdown period. It was a unique period because at that time, the beaches were closed to people, and we never had that before. We never had a period of time where months elapsed where the beaches were closed. But we set up the study because Key Biscayne asked us to look at their beach to find out why their beaches kept failing to close consistently. And so we designed a sample collection program. Immediately when we visited the beach, there was a brown area along the intertidal zone, and immediately in my mind, see this, that brown zone. It's a zone where they were taking sargassum and breaking it up and integrating it into the sand. So our sample collection plan, of course, includes the waste, meat, and ankle deep water. It includes our different zones from the sand. And in this case, we added the seaweed. So we intentionally, in this particular case, looked at the sargassum. In this study, again, we measured for enteric oxide by membrane filtration, and then we also added in superimposed microbial source tracking markers, and these are specific organisms that we look at to evaluate the influence of human fecal waste, bird or gull fecal waste, and also dog fecal waste. So this is what the beaches look like before, during, and after the shutdown period at the beaches. Of course, we see the people before and after, no people during, and again, we collected our samples in the various zones. And what we found for the water is every time that there was a sargassum inundation, either during the inundation or shortly thereafter as the sargassum was decaying, we found high levels of enteric oxide in the water. In the sediment, again, that mystery that we didn't know why the white sediment at the top, the supertidal sand, had high levels. Well, we solved it during the this study. If you see, when the beaches were open, the supratidal sand, which is the sand that typically is contaminated with bacteria, is high. But then when the beaches were closed, the supertidal sand dropped. So there was something about the presence of people that influenced the amount of bacteria in that zone. The integrated sand, in both cases, whether or not the beach was open or closed, remained high. And so what we assumed, or what we believe is going on, of course, the zone where the bacteria really are growing and accumulating is in the integrated zone, where they're mixing the seaweed with the cement. But we believe that the human presence is the dominating factor for the spread. People go back and forth from the water to that beautiful white zone, the supertidal zone, and they carry with them their water, they carry all the sand, and they walk through the seaweed and get up there. And so the people are the humans are the ones moving around. Ice levels in the supertidal. We also looked at the seaweed. The seaweed also had elevated levels of bacteria. We found a lot of gull marker in the seaweed. We believe that the fecal indicator bacteria there are partly making the gulls, and during the closed period, we saw a dominance of gull marker within the beach. Then we believe that the seaweed provided an environment for growth, and higher bacteria was associated with the seaweed. So that brings us to our current work, where we are expanding. We're doing a project somewhat similar to project number 1, but now we're adding in the sargassum. We didn't realize the importance of the sargassum to the overall quality of the beach. And I'm collaborating with Dr. Jai Lee of Mechanical Engineering. She's leading the project. We're looking at emissions from sargassum. She's leading sulfur and microbial emissions. And we are learning, I am learning a lot about aerosol sampling in terms of tubular bags, and now we're using impingers, which are now, those samples are now being used for sequencing analysis. So with that, I'd like to acknowledge prior studies, Dr. Milani, predominantly for work, I agree with Dr. James Klaus, Sandra Gaston, master student Peter Solwell, Chris and Mary Beth from NOAA, who did the microbial source tracking, and then the current active research project Dr. Lee, and her team, and my PhD student Brittany and Grass. With that, I'd like to 15
Head over to the next one. Oh, that's great. Gosh.
Thank you very much. So let's take a moment to thank all of our speakers again for some really great talk. Now we're going to get to our panel discussion. So if speakers could come up to the front. I'll bring you some chairs.
How long is it? It's gonna be 4:30. Okay.
Now,
All right, so one thing that Young and I were talking about today was seeing if we could get one question per table. Just to make sure that we can get a presentation of not just faculty, but also students as well, asking questions. So I'm gonna start over here because I'm gonna pick on my group. All right, so any question you want to ask either to all the panelists or to one specific panelist. Go ahead. All right.
I guess this is targeted more at the Catalyst team, but anyone, when you're trying to get the community involved in science, what's usually the biggest barrier that you see to getting that involvement?
Great question.
Thank you. Well, some of what I said, right? The information is really hard to get to, hard to understand. So working with colleagues who understand the science and who can share it with us in a way that we can sort of demystify it and make it like more accessible, more relatable. And then you have your usual barriers, you know, language barriers, work schedules, need for childcare, dinner, things like that. So we do our best to address that, you know, at the front end, you know, by offering food and interpretation and childcare and things like that, and then organizing things, events, either online with people who are able to do it online in a hybrid setting, or after work hours or on a weekend and a place that's accessible by transit so that people can get there.
Tackle that question as well. So any community engagement that's been done and any sort of challenges that you faced during your studies?
We work like Key Biscayne. We work with the village of Key Biscayne. We also have projects in Miami Beach. And the community, usually by the time they ask us to come in, the community is just very upset. And they come in with, you know, preconceptions. And yeah, it's just difficult to, there's a lot of, sometimes they just are suspicious and they think that maybe we're not in the same side. You know, we're trying to find the answer, and they sometimes feel as though, yeah, sometimes we can't find the answer, and it's hard. And so what is the solution for, let's say, Key Biscayne was an easy one. It's an easier one because we did find a solution. You can't integrate. You're not supposed to be integrated. But sometimes we can't find a solution. We don't know. And then in that case, there's some frustration from the community. So I think that's, to me, a challenge.
One thing I can talk about for the Algolon work we've been doing, we're part of this larger center I mentioned that just got started last year or a year and a half ago. And we have been, some of the members of our center have been working with some of the people running the fishing boats out of northern Ohio and they have been for many years. And I think our team makes a very conscious effort to engage with and not just sort of, you know, say, well, we see this, you need to do that, but more so kind of, you know, they're having asthma-related issues, they're having, you know, health things, they want to understand, but they also don't want their lives to be impeded. So it's finding that balance. And so, but I think it takes time, it takes building that trust to be able to be effective that way. And I think it takes long sort of partnerships to typically be able to be particularly effective.
Lauren?
So I haven't personally done any of that myself, but NASA has an interest in making their data more actionable, and they actually have funding opportunities now to do that. And part of what they're trying to do is have an actual person, like community partner, like they have to be a co-I on the proposal, and they have to have an idea of what they're going to do, and they need to be able to take over after the NASA scientist comes or it doesn't even have to be NASA scientist, the scientist who got funded, you know, works with them to develop a product, and then that scientists would no longer be funded to work with them, but the hope would be that that group would then use that product. But we don't necessarily know what people need. So I guess the trick is finding people who need our data and you can find, and then them finding us. That's the tricky part, is finding each other.
Great, thank you very much. And thanks for being the first guinea pig. All right, question from this table.
Dennis, question.
Oh, all right. I'll come back to you. I won't forget.
Thank you all for this. What I'm about to ask is more in relation to what he asked. And let me give the disclaimer, I'm not political. So a lot of talk goes into policies. And one thing I've come to realize is that, yes, community engagement is a very necessary part, but then we fail to think about the political aspect. Because, they are our biggest stakeholders. So what do you do, or how do you navigate when your biggest opponent is more political than community engagement?
That is an excellent question. I'm gonna let Camilla take the lead on that one.
If I were a magician and had a magic wand, I'd get money out of politics, for sure, because that's the biggest issue. But no, I think that involving community members and then making the information accessible so that they can use their lived experiences and their voices to advocate for change is a huge first step, because normally we're locked out of the process, decision-making process that should involve community because it has a direct impact in community. And we don't really have that. We have a very low level of civic engagement in general in Miami-Dade County when it comes to, for instance, local elections, and other issues that have direct impacts in community. And I think that requires not only a change in the political class, but also within community. And I'm not blaming community. I know that there have been many examples, when either scientists or companies or nonprofits or governments have shown up at a community with promises that do not get delivered ever. And so that creates distrust. So, working with a community partner that has rapport and has built trust with community members is a good way for other institutions to be able to make inroads. And I think that building that community muscle to go with the advocacy is going to be hugely important for any policy, whether it's environmental, housing, healthcare, or anything else.
Yeah, I agree with everything Camila said. I would say for the Grand Lake St. Mary's Storm Balloon project I was talking about, that's right. There's a Wright State University satellite campus there that's maybe 2 or 3 buildings that one of the professors there, Stephen Tuckerman, that we work with, he's been trying to trying to get plants essentially into the lake that are going to take up some of the nutrients to help reduce how bad the harmful algal blooms are. And kind of watching how he's gone through that process, I'll be political, it's a 78% Trump county. Like, we're not, a lot of the scientists there are not viewed as necessarily particularly welcome. And what he's done is there's certain people that are just never going to work with him, but there are people, even in that community, that are willing to work with him, or at least, have a conversation, and he approaches it very apolitically. He tries to, find common ground in a way that's not going to cause people's defenses to come up. And as he has made progress with doing that with the few people that maybe were a little more open-minded, that has started to sort of break down some of the other barriers of other people. But it's a long process. He's been at it for 8-10 years, and he's making some progress, but it's slow, and it takes a sustained commitment, which often on our 3 to 5 year funding cycles can be really tough to maintain. So I don't mean to give it a Pollyanna-ish answer, but I think that's at least a model that has the potential for success.
Yeah, I agree. Keeping the politics out would be the best of all worlds. In terms of the Miami Beach project, what we do is at the end of the project, we do a public presentation, and we have community members come and provide feedback and input, and then we incorporate their questions and answers to their questions as part of that, as a way to address community concerns. But in some cases, they come in with expectations that you can't necessarily meet. And we never really made a promise to begin with, but
they have an expectation the problem's gonna be fixed, and these environmental problems are sometimes, they're so complex that, an easy solution is not the answer. Thank you for the question. All right, I'm coming back, like I promised. Someone do it, otherwise I'm gonna pick someone.
Hi, my name is Mahir. I just have a question, like, for what would be your advice for a young, budding, like, scientist in the field who wants to continue the research, but also, like, navigating the path to get the right funding or to apply for the right funding? Because right now there are so many different funding agencies, but the niche is getting very much like smaller and smaller nowadays. So how would we target that, and like, what is the best rate? Like, I would not say like, what is the success rate where we can get 100% sure that we can get it, but still, how we can outstand there so that like we have higher chances, or how we can plan that?
Great question. I'm gonna take notes because I want the funding advice too.
There's no way, I wish there was a simple one-size-fits-all answer. I think the commonality I've seen between people that end up being, that are often pretty successful, is that they learn during grad school about a broad range of topics, and they have the adaptability and flexibility to follow where the trends are going to be. I can just give an example. What I had in my faculty proposals when I was on the job market has 0% overlap with what I'm doing today. And within 2-3 years, that was true. But I was fortunate to be in a situation to be able to connect with different people. And so that's one of these workshops. I'll give a shout out to Cassie and CY and everyone. It's meeting people at these workshops, coming up with ideas. And frankly, partnering up with more senior folks that often are more established, that's a great way to break through that, because when I was putting proposals in early on, none of the funding agencies knew who I was or what I was trying to do. But if I partnered up with a more senior colleague, that was often a great way to get started. But being adaptable, having a broad base of knowledge to be able to follow different things. If someone says, I just do, if I had, for example, just done secondary aerosol and not had this other interest or these broader things, it took, the way I presented it today, it sounded like we had this cohesive plan over a decade, right? But it took us 6 years before we got any funding to do any of that. That was all kind of bootstrapped on the side from like little pots of money here and there before we got to the point of having enough, sort of established to be able to get funding for it. So sorry if that's not a particularly helpful answer, but that's at least how I think about it.
Yeah, I agree. What I would recommend is, faculty who I've seen succeed typically will be able to use what they're good at, their background, but then they leverage that. Going completely into something new usually doesn't work if you go too diverse, but you want to use your background to leverage something new as well. I also recommend that there's a lot of different, like you mentioned, there's a lot of opportunities out there. There are opportunities that have higher probability, usually there's smaller amounts of money, and then there's the big ones with the big risk. And I recommend trying for all of them and being happy with getting the little ones, because those little ones help get you the big one. And it's like, I see it, you gotta get the ball rolling, and then once it's rolling, it's easier to get the larger ones that are higher risk.
Yeah, I agree with what everybody has said so far. Another thing that helps is having the right team. So don't pick your friends per se. Not like that, but like if you're going a little bit outside your field, you want the expert in that field, not somebody that necessarily you worked before, but in a different field. So team really matters. Getting feedback is really helpful. And early, it takes a long time, a lot longer to write grants than a lot of younger people think it's going to take. So start early. And then ask for feedback before, maybe 2 weeks before it's due, like talk with other people that have them actually read through it and give you feedback and then respond to that feedback. That helps a lot. I had a list that I gave the students. I'm trying to remember what those were. I think it really helps to be on point with what the grant is asking. Like the different agencies are sort of different. I can only really speak mostly about NASA, but they're top down. So it's not like, I think NSF is more bottom up. Like you come up with cool ideas and propose them. NASA has they have the ideas that they want to do, and they're saying like, who can help us do this thing? And so you don't propose what you want to do, propose what they want, and that helps a lot.
Yeah, I fully wanted to build on that point just slightly is that, yeah, essentially NSF and NIH to a degree are give us your best idea and, you have to find what's the right program. So my two pieces of advice, one is that if you were going to NSF or NIH, make sure for NSF that you talk to the program officer ahead of time. I know a lot of folks will spend all their time writing a proposal, send it in without having to talk to the program officer ahead of time, who might have just said like, hey, that's great, but that doesn't fit my program, or it doesn't quite fit what we're looking for. But usually if you say have a one-pager and you set up a time to have a talk with them, either at a conference or over the phone, they're never going to say, oh, we can fund this, but what they will say is that's definitely in the scope of what my program funds. And that gives you better, so for the bottom up ones, that's the way to do it. And for the top down, which the more mission driven agencies like NASA was being mentioned, but also really every other funding agency I can think of beyond NSF and NIH, you need to find a way to do what you want to do within doing what they are asking you to do. So you figure out what they want and then figure out a way to do that in a way that's interesting and fun for you. That would be the advice. Because if you just propose to DOE or NASA or NOAA or something, things that you're like, oh, this is really interesting, they're gonna be like, yeah, but that's not what our mission is, what we're trying to get done. And I had very low success rate, and was really on because I didn't learn that lesson early.
Yeah, same. I was going to say, for those of you who are interested in that topic, it's a good topic to think about. There is a workshop series that's happening on grant funding. So the first workshop is going to be on setting up the timeline, like how you actually set up the timeline for writing a grant. The next one is going to be on diversifying your portfolio. So if you missed that email, or if you're not from University of Miami, let us know. We can send that to you. And I highly encourage all of you to think about that, because attending those kinds of workshops can really be a game changer. It helped me because I didn't know what I was doing when I started this job, to be perfectly honest. All right. Any questions here? I'm gonna put you on the spot.
I'll give the opportunity to students.
Spoken like a true PI. Excellent.
Well, I can ask a question. Maybe we can get back to the community engagement. A lot of the students here are engineering students. Of course, we also have the students from natural science. But say, for these students, during their education, what would you recommend they should do so they can be better prepared to do something related to community engagement research?
Fabulous question. Right, let's just start. Milo?
Great. I would say talk to your professors who already are involved in partnerships with community partners so that you can be a part of that and help connect the science to the reality of our communities. The air quality monitoring stations at the county, they need engineers. The person running that is an engineer. So you may look for internship opportunities at the county. We're also in partnership with the City of Miami, Office of Sustainability and Resilience. They do a lot of air quality work, also water quality. We are doing a lot of work with extreme heat, installing sensors in people's homes, monitoring that, and then bringing people into the discussions that may lead to policy changes to better address extreme heat. So there's a lot of stuff going on out there, and usually it happens in partnership with community organizations because of what some of my fellow panelists have said, that if you haven't built rapport and trust within the community, it's going to be a lot harder for those scientists and those government officials to carry out the project by themselves. And so I think that there's a lot of opportunity for partnership between academic institutions and nonprofit organizations on the ground. I think it's just a matter of finding the right project and then talking to your professor about reaching out to those CEOs or community-based organizations.
Yeah, I completely agree with what Camila was saying. I'd also say that not just UPIs, that there's often programs where you can be able to do outreach potentially as a graduate student or an undergraduate. I know, Cassie and I were in grad school. We often had outreach that our advisor had organized with different schools in the area. And there's a lot of community organizations that also, anyone that has an NSF grant often has a broader impacts component, which is going to usually involve some portion of the community, nearby. And so those are great opportunities to get practice at, sort of engaging in a way that's going to help you to be able to build those relationships then when you are in a position to be doing that yourself. And so if you start somewhere new, I know anyone that comes to Michigan, for example, there's already existing structures in place that you can get involved with, and as you meet people through those, you can then branch off and do your own thing as well. But there's a lot of infrastructure in many places, including Miami, where you can start that process. You just kind of have to ask around until you find people who can be able to help you out with that PI or beyond.
Yeah, I'll also I would just say, being proactive as a grad student is really helpful. So, I had my first student was really proactive about pushing me along with sort of expanding our collaborations with the project that we were doing, and it really changed the direction of research in my group. So don't be afraid to talk to your PI about, bringing your own ideas to the table about how your lab in general can be more engaged with the community, because it really could shape and enhance your science as well as a side benefit.
Hello, my name is Christy. So this is a collaborating question for all of your areas. We were wondering, in regards to each of your areas, what are certain gaps, whether that's either resources or lacks that you see happening, or also knowledge questions. What are things that you're seeing? What's next? What's coming after everything that's been done so far?
This could take a while to answer. That was a good one. Anyone want to go first?
It's not really a gap. I think I did work in microbiology, and I found that during the COVID pandemic, the whole world came together to learn new techniques and new methods, and we have advanced the ability to detect and measure things in the environment in ways that were unimaginable before the pandemic. And I feel as though now people are implementing it, coming up with new questions, and now we can see things that we never saw before. So I feel as though we're now taking all the information, the advances that we made during that period, because it was not only the environmental scientists that came together, but it was the medical community that had incredible expertise when it comes to microbiology. Put it all together, and now we have these tools for environmental forensics, environmental engineering that we never had before. And so I feel as though the door is wide open in terms of using these new methods for looking at not only where the contamination is coming from the beach, but we can even use wastewater to analyze disease in communities. We can do a lot more now.
There's been a lot of really cool stuff happening. One new thing is that there are a lot more small stats than there used to be. There used to be just these really big missions, and that's all there was. But now there's a lot cheap. We can do things a lot cheaper and measure a lot of different things. And it's not just NASA's the only game in town anymore. There's tons of industry groups that are now taking over what we used to do. And so now there's so many more opportunities in space science in that regard. Machine learning and AI is, of course, a really kind of untapped and big resource. Yeah, those are just two of the things that kind of pop into my mind.
Not to get very political, but in this new political climate, to put it some way safe, a lot has changed before this administration took over. There was a lot of money coming down from the federal government through the Inflation Reduction Act, through the APA, NHS, FEMA, NOAA, you name it. One of the concerns that we had was how are we going to ensure that we have built the right infrastructure at the ground level to ensure that the the perpetrators of the environmental degradation are not the ones actually gobbling all the funds, but to ensure that some of those funds actually get to community organizations where communities have been disenfranchised and marginalized historically, to a situation where pretty much all of that stopped. And not only did it stop, all the money coming down, but because of the layoffs and all the staff adjustments, to put it some way, now there's no technical assistance, there's a lot fewer staff to help with implementation. And so, for instance, the work that I mentioned that I was doing before, providing coaching for grant writers, that was paid for by the EPA through their TIC-TAC centers, which were basically technical assistance. That went away. And so small size community organizations that don't have writers on staff, or development, or policy, or anybody who's able to stay on top of policies and who knows how to write a competitive grant application, don't really have a chance to apply for a lot of these opportunities. And then also, because of the reduction in staff at these agencies, there are fewer people who are dedicated to helping those community organizations or to ensuring that those programs are implemented or even the funds dispersed. The other issue is that we're seeing a lot. We work with undocumented community members, and there's a lot of fear. I mean, I'm sure you have seen the anti-ICE protests, and that has a real impact in organizing and community advocacy because people are afraid. They have passed a lot of laws, even before the mass protests, where they have basically deputized local governments to do immigration work. And that has sent chills down the spines of a lot of community members who used to be active, used to come out and be civically engaged, and no longer are doing so because they're afraid. So those are some of the gaps and the issues that I'm seeing with the funding, but also with the level of engagement.
Yeah, no, I'll agree. I would say that it's interesting in the environmental world. I mean, I think that I'll be candid. I don't really care about the politics of it, but the environmental work in environmental areas often goes in somewhat cycles or waves where there will be a supportive administration and then potentially less supportive administration. And, but we haven't quite seen the straight up attack on science that we've seen recently until this past year, of course. But in those times, I think where a lot of the people who've been very, again, successful kind of in the community, what they are able to do is to both shift gears, look more locally, look more, if you have a supportive state or at the statewide, look at nonprofits. Those are all places where you can essentially try to come at a problem in the same way we were kind of hearing this morning from Holly a little bit. How do you approach something in a way that isn't going to cause people's defenses to go up instantaneously? How do you frame questions? And there's always going to be questions that can be framed that way, but it often takes a lot of work, some creativity. But there's also, I mean, to go to this one, I mean, there's a lot that's also just not going to get done. I mean, it's all, just to be straight up about it, like there's stuff that isn't going to happen that we need to have happen. And so there's, I think, a great thing, the number of, at least I've seen students going through our program over the years have done, is to go into policy or go into politics to try and actually make a difference. Because, if you don't have anyone, I think there's less than, I think I can count on one hand the number of scientists that are currently in Congress. And so, not saying any one of you needs to necessarily, be in DC or something like that, but that is the way that people can try to make a difference as well. Just staying out of the game doesn't, fix things.
All right, we already had a question from here, so can thank you for that. Any other questions? Holly, you want to answer the question? Put you on the spot. All right.
Well, first I have a specific question for Lauren, and then I was trying to make it into a general one for everybody, but I know in my community, everyone's upset with this, ready to go offline and clean up life. And Modus, and just thinking about the budget cuts that have happened, the staff cuts that have happened, satellites take years to develop, sensors take years to develop. Should there be data sets that we're concerned about or that you're concerned about that are going to not have a predecessor in the next several years? And then I guess for everybody, personally, like the last year between
the freezing of the federal funds and then government shutdown, I actually received a pause work order, which I've never had before, from a government shutdown. And like NASA Roses, a solicitation that goes up for everybody. If you don't know, they call it NASA Roses. It comes out on Valentine's Day every year, except last year it didn't come out for months. And then it was about, what would you say, a tenth of what it normally was solicitation-wise. How are you reframing your projects and your work in a different way to make it to where you're conscious of, oh, I might have to stop this in a month, or I might have to shelve it, and how do you keep from losing momentum?
Fantastic question. All right, Lauren. Okay. To answer your first question about Modus, so Modus does have a successor, which is Beers, and there's no plans to shut down Beers right now, so that's the good news. Also, they're trying to actually sell off Modus, so the hope is that maybe someone will buy it and keep it going. Somebody who might use those data, and they're preferentially hoping to sell it to somebody who will keep the data open access. So we'll have to see how that goes. But anyways, Modus-like products should continue, and that's actually always the plan. Like Modus was going, no satellite does forever. It's already been up since like 2002, so it's pretty old right now. So that's the good news. Regarding resources that we're not going to have anymore that we should have, absolutely, we don't have a LiDAR. The LiDAR is really, well, the Europeans have a LiDAR. We don't have a LiDAR. We had this big plan for this really cool system, and they kept reducing the amount of funds. It's called ALS, and eventually we discanned it. And it's unclear whether we're going to even be investing in large-scale Earth science missions. Probably, I don't know. I can't speculate, but I suspect maybe in this administration, maybe not. But maybe in the future we will. But like I said, there are smaller, now it's cheaper to do other, like small sats and things like that. But so there are different, maybe going the same way we have been going isn't the best way forward. Maybe we need to just adapt, go on these cheaper missions. In the meantime, we do have the EarthCare, which have a LiDAR. The LiDAR, if you guys remember, that's the vertical profile information. That's really critical information. So it was a really big blow to NASA that it didn't come out. But maybe in the future, we've done a lot of work preparing for it. But these things do take a long time. Like you said, it takes a long time to build. And your third question was, how do you, Roses came out late last year. And I can't, I'm not an NASA official. I can't speak on behalf of NASA. This is just my personal opinion. I'm personally not trying to rely on those funds anymore because it's a little bit unclear what's going to happen. I mean, you can't bank on something, when, it's not consistent. Hopefully we'll get it all sorted in the future, and it will be, but for now I've sort of been trying to diversify. So I've gotten funding from several nonprofits. So I'm doing work on actually climate intervention. So that's actually something NASA doesn't want to fund at all. Because it's a little bit like political, it's not their thing, but it's looking at the topic that I got funding to look at is whether, I remember I showed some images of how clouds were getting glaciated and then they changed their properties. So there's this idea that if you glaciate clouds in the Arctic or the Antarctic in the winter, you might cause those clouds to kind of the hypothesis is you could glaciate them and then make them disappear, and they act sort of like a blanket warming the Earth. And so if you would disappear those clouds, maybe you could keep the sea ice. But this is all very hypothetical. So they need scientists to see, could this work? Before you even say, is this a good or bad idea? First of all, you need to know, is this even worth studying? And so I pivoted a bit towards that direction. I don't know, other people might have other ideas, but I think I realize it's good to diversify. To answer your question.
Yeah, I agree. Diversifying is important, especially in these times when you have a roller coaster. And this last year has been a roller coaster in terms of the funding agencies being cut through DOGE, and then we had a government shutdown as well, which postponed things. But through all of that, during the intense part of it, sometimes you question yourself, but you always have to keep trying, you always have to keep going, you have to find a way, you can't give up, then you're done. So yeah, keep applying and keep submitting grants, maybe not to the traditional agencies, maybe looking at some other opportunities. I know that they've been circulating foundation opportunities, non-governmental. So, and there's a lot of internal opportunities at the university as well. And you just don't, as mentioned, look at these other opportunities in diversity.
We have seen a cut in funding, not us particularly, because we were very diverse in our funding sources, but we don't take any federal money. But we did, we were co-applicants for a community change grant, an NHS grant, etc. And we're not relying on it. We don't count on money unless we received it, so we're okay. But we have seen a reduction in funding, whether it's because it's completely frozen or because the money was approved but it was not given to the institution. As far as the rebranding, absolutely, especially when one of the applicants or one of the partners is a local government, especially in this political climate, in this particular state, but I would say more generally as well, where particular projects have had to rebrand the name in order to stay away from words that could be inflammatory to the White House occupant or different government agencies. And so that's a real thing. We are not rebranding ourselves because, again, we don't have any federal funding funding our programs and our initiatives. But when we have a campaign, for instance, that's built around racial justice or gender justice or environmental justice and things like that, we communicate with the foundation or with whoever's providing funds to ensure that they're okay with our continuing to using the same narratives that we have used. So we personally are not, we at Catalyst have not changed anything if it has been up to us, but we have seen it in larger coalition work.
Yeah, I think the panelists all made some great points. I mean, I think, I at least give an anecdote of what we dealt with as that center grant. I'm not sure that we have that's doing a lot of our work. We had to go through in September, October of last year and scrub the word climate everywhere out of the grant and resubmit it. And that's not uncommon. I noticed the folks that applied to DOE that had to do the same thing. So I think for those things, if it's able, if something like that lets you keep the grant, then you do that. And the main ideas of what you're trying to do aren't going to change that much. So you can stick with those. I think where I'm more nervous, to be candid, is that I think a lot of things are kind of wrapping up this year, next year, the things that were, had already started. It's a lot harder to stop something that's already going than to just not do more. And so, in terms of getting rid of some of the programs and part potentially having the issues that, we've heard about with that, National Center for Atmospheric Research, I think in those senses, I'm honestly pretty pessimistic. So I think, as everyone has said, kind of going to more local sources, now is the time that if you want to try something that doesn't follow in the more traditional ways that people have been funded. For example, we do a lot of secondary organic aerosol work right now. I'm submitting way less on that than I typically have because I just realized that's not likely to get funded. But some people microplastics are out right now, so we're putting in more on microplastics. So just kind of figuring out where people's interests are going to be both on the nonprofit but sort of state and local side of things. Sorry.
Yes, I didn't mean to cut you off, so sorry. I just had another thought, which is that there are commonalities between administrations and focusing on those things might be good. Like people tend to agree that wildfires are an issue. Nobody wants their city to burn down. People tend to agree that food is good, so agriculture, things like that. So there are some commonalities that those might be good areas too to kind of consider.
Yeah, and again, just to end on a little bit more of a happier note. No, it's fine. Again, there is those workshops to help diversify portfolios. There are resources here that can help. Also, for those of you who are students or postdocs at UM, we have a writing center. It's really good. I use it all the time, and it has really helped me improve my funding success rate. Highly encourage you to all use it. It's free. And they actually have pretty good know-how of science writing. So any of you who are thinking about writing papers, proposals, even just, something for a class, take advantage of your resources now because it's really going to help pay off for opportunities for future. All right, any other questions before, so John, all right.
So in the recent years, we've had a lot of new interest in machine learning and AI. I wanted to ask, how have you guys already incorporated or plan to incorporate that in your research, funding, writing grants, as well as mentoring students with AI and machine learning?
We actually we brought in an organization to do a training for all staff on the different ways that artificial intelligence can be used in the workplace. So starting with us, we're starting to implement certain things that make the work easier. We have been doing that all along without even knowing that, because AI has actually been a part of a lot of systems that people just didn't know about. But yeah, we have been learning about that, and then the next step is going to be to cover the technology gap with all these new technologies that are coming. This is something that happened first with the pandemic, where a lot of the work that community-based organizations were doing in person had to start doing online, and a lot of people didn't have the access to Wi-Fi or the know-how for different platforms like Microsoft Teams or Zoom or Google Meets or anything like that. So we engaged in the process of teaching community members how to use these technologies so that we can continue providing the services and bringing people into programming, even if we could not do it face-to-face. So now with this whole new tsunami of new technologies, we're also thinking of ways that we can bring community members into space where they can learn the different applications of the new technologies as well.
You also mentioned, in addition to AI in our day-to-day use, you also mentioned machine learning. And I do think that machine learning is also making major impacts in how we analyze our environmental data. In the past, sometimes it's hard to come up with relationships, and machine learning is one of those ways in which we can see what's related to what else and come up with models. I don't see machine learning as being to me, I get a sense of a black box, and I don't see it as a substitute for a mechanistic understanding of how the system works. But I do think that the output of the machine learning can be used to better understand how it works. So I think they would be complementary to each other as well. So yeah, I do think that machine learning will revolutionize also how we analyze data, and it's going to be another tool that we use to supplement.
Yeah, I think it's great. There's a lot of colleagues that I work with who use it, and I don't use it myself unless you call like some of the statistical methods I use, but I'd love to. I just haven't yet. I'd love to hire somebody if I had money.
Yeah, I think, I'll try to not ramble on for too long, but I think it's a tool, as was just said. I think there are places where we can use AI and machine learning, and we have a ton of data off in the environment. So there are places where we can absolutely find associations that we wouldn't otherwise if we use it in an appropriate way. And we've done, I feel like there's a little bit of public consciousness, this idea that somehow this is all brand new, where really we've been building towards this over time, I mean, you're busy, some of these clustering algorithms, right, that were machine learning before machine learning. So it's a thing where I think, obviously, the growth in particular aspects of it has been very fast. I think both a cautionary note and sort of a broader picture note. A cautionary note is, I will say, there are going to be people that are going to misuse it. I know we recently got reviews back on paper where somebody had plugged it into ChatGPT and spit out a review for it, which is against every ethical guideline the journals provide, but that's going to become more and more common. So being able to identify that, I think is going to be a thing. And then I think as a broader picture note, I'll try to put a positive spin on it, is that I think, at the end of the day, AI and machine learning, they are trained on a data set. That data set is what is already exists, right? And generally, most folks in this room, a lot of what we're trying to do is create new knowledge. And so it's not trained on anything that doesn't exist yet. And so being able to think creatively and to be someone that isn't just plugging into what we already know, to be able to think of new angles and new directions, I think is where we still need people, you know, and we don't want to, but I worry a little bit that it's going to drive science in certain directions based on sort of, I think it'll make big things tougher sometimes because people will focus in on areas that are already pretty heavily studied that will make it more incremental in terms of progress. But those that can get beyond that, I think are going to have a competitive advantage.
Great, so let's thank our panelists again. Lonnie is going to announce some of the poster awards as well as provide some closing remarks, so stay tuned.
Okay, so we had a wonderful poster this afternoon, and now we are ready to announce the awardees. And for that, I would like to have Aaron coming over here to give awards for our third place winners. And the third place winners, we have two of them, and actually they spread across all the different institutions. So the first one, third place, Mahendra Rawat. And also Tahilit Boris from Miami. Tahilit Boris. I can kind of take a photo. What about again? Wait a little bit. So we have all the yeah. Okay. The next one I'd like to have Aretha from EI for the second place, and it says the winner is Rena Jackler. Okay, now the last one, the first place, and I would like to have David Hagen to give an award. Okay, so who is the winner? Kazi Tashina Habib. From US, right? Yeah. Okay, now we're going to take a photo together. Congratulations again. And so this is the end of the workshop, and I hope you enjoyed it. We have covered different topics and with a very diverse perspective. And I hope the interface can brainstorm, stimulate some ideas that you can work together. And hopefully we will see you next year here again. Thank you very much for coming. Thank you.
Yeah, thank you. Okay.